Hello, everyone, and welcome to the third annual Frontiers in Ocean Sciences Symposium, organized by the Division of Ocean Sciences of the Director of Geosciences in the U.S. National Science Foundation. Just a couple of quick notices. Audience members can submit questions to the speakers via the questions and answers box. Otherwise, chat will be turned off during the symposium for the audience. This symposium is being recorded, and the video will be available after the meeting with a link sent to all registrants. But without any further ado, I shall pass to Clea Harrelson to introduce our first speaker and the symposium's moderator. Clea is a Knauss Fellow in the Division of Ocean Sciences, working on a number of science and policy issues. And as an early career rising star, it's very apt that she introduces our opening speaker for this symposium on the future of ocean sciences. Take it away, Clea.
Thank you so much, Chris. It's my pleasure today to introduce you all to Dr. Terry Quinn. Dr. Quinn has served as the NSF Ocean Sciences Division Director since 2018. He is on leave from the University of Texas, Austin, where he is a professor in the Jackson School of Geosciences. Dr. Quinn served as the director of the University of Texas Institute for Geophysics from 2009 to 2018. He completed his undergraduate degree from SUNY, a master's from Wichita State University, and his PhD from Brown University. Following a postdoctorate at the University of Michigan, he was promoted to the rank of professor at the University of South Florida in the College of Marine Sciences before moving to Texas. Dr. Quinn's research interests focus on paleoclimate and paleoceanography. He has written or co-authored over 75 peer-reviewed articles, many of them with his graduate students. He was a lead on the author of the fifth assessment report of the IPCC, and he has served on several executive committees of the International Ocean Discovery Program and participated in 3 International Ocean Discovery Program expeditions in his career. We are very grateful for your time here today. Dr. Quinn, please take it away.
Thank you very much, Clea. It's a real pleasure to be here with all of you today. So I want to thank you all for joining us, like you said, the third annual National Science Foundation's Frontier of Ocean Sciences Symposium. We are so thrilled to share with you today. We have 4 early career scientists from universities across the country. They're going to share their research, their stories, and their thoughts about the future of the ocean. We also have a panel of last year's speakers, and I am looking forward to hearing updates on their research and to engaging in a lively discussion with them and you all. Today's speakers are at the frontiers of ocean sciences, and their forward-thinking and transformative research are key to advancing the field and helping us to understand and adapt to our changing oceans. The theme of this year's symposium is the future of the oceans. One of the main questions we'll be asking is, what will the oceans of the future look like? In addition to this, we'll be asking, what will the ocean science of the future look like? And importantly, who will be the ocean scientists of the future? And how can we support them and help them realize their full potential? But before we talk about the future, perhaps we should look back to the past and how ocean science and ocean scientists have changed during the history of the National Science Foundation and even during my career. NSF was established in 1950. 4 of the NSF's first 100 awards were related to ocean sciences. So NSF has been a leader in basic ocean science since its establishment. But it was a very different world then. In many parts of the US, schools, restaurants, and public transport were segregated. Oil was just starting to take over from coal as the world's foremost energy source. Fish were seen as an inexhaustible resource in the oceans, and there was an active whaling industry in the Antarctic. The US had just come out of the Second World War, and many in the ocean sciences learned their trade in the military. With a few notable exceptions, ocean scientists were almost exclusively white men. In addition, a great deal of ocean science data and information we now take for granted were considered to be a closely guarded secret. The closest thing to a personal computer was a 4 cubic feet in size and had just 2 bits of memory. In the 1950s, in my field of the marine geosciences, some of the basic concepts of the field, like plate tectonics, seafloor spreading, and continental drift, were considered to be controversial ideas. Indeed, the groundbreaking marine geologist and underwater cartographer Marie Tharp's revolutionary ideas on seafloor spreading were infamously dismissed as girl talk. Marie Tharp herself wasn't even allowed to go on an oceanographic expedition until 1965, when she went on the NSF research vessel Eastward, which was the only ship allowing female scientists on board at the time. However, over the past 70 years, not only have the state of the oceans changed dramatically, but the nature of ocean science and ocean scientists has likewise changed drastically. Even in my lifetime, how ocean science is conducted has changed tremendously. When I was starting my PhD, which to me now feels like the Pliocene, but a cutting-edge computer had just 64 kilobytes of memory. The National Science Foundation's NET had just been proposed, a then innovative idea about having a network of computers to share scientific and educational information. The internet did not exist. There was no such thing as email. Mobile phones, if you could afford one, weighed over 2 pounds, took 10 hours to charge, and ran out of power in just 30 minutes. But some of the first ocean observation systems were also being deployed in the mid-1980s. To measure ocean temperature, scientists were starting to become concerned about the changes in global climate. 35 years later, just as the iPhones in our pockets today are descendants of the Commodore 64 computer and 2-pound mobile phones, the recently funded Global Ocean Biogeochemistry Array, or GOBGC, stems from this early ocean observation systems. The new systems will transform the way we observe the global ocean, deploying 500 floating robots around the world, collecting important biological, chemical, and physical data from the ocean surface to depths of 2 kilometers. This system will provide essential data on how climate is changing the ocean's chemistry, biology, physics, and geology. When I was a PhD student, the idea of having a fleet of robotic floats automatically collecting ocean chemistry and geological data throughout the water column, and then beaming these data wirelessly to a home computer so that they could be analyzed was pure science fiction. Work then would have required labs full of scientists, each with their own most decidedly not portable computers. What will it be like collecting ocean science data 35 years from now? Many of our speakers today are likewise looking at how climate is changing the ocean. For some, it's on a historical scale. For others, it's changes in ocean chemistry and physical process, or the impacts of climate change on marine organisms. Hopefully, in 35 years' time, the current speakers will be reminiscing to their students about how, when they were early career scientists, the climate change threats they studied were ably addressed by policymakers, by industry and society, and a growing crisis was averted. Now, let me speak more to our planet's current greatest challenge, climate change. Climate change is a crisis, and one inherently linked to the ocean. As Secretary Kerry often states, you cannot address the climate crisis without the oceans, and you cannot solve ocean issues without addressing climate. President Biden has declared climate change to be the #1 issue facing humanity. And as such, tackling the problem of climate change is one of the administration's 4 pillar priorities. NSF has played a vital role providing funding, instrumentation, and infrastructure for the identification of climate change impacts, in addition to investing in the scientists and students who conduct this essential research. 3 ways that changing climate is fundamentally changing the characteristics of the ocean are ocean acidification, ocean circulation, and rising sea levels. As the ocean absorbs more carbon dioxide, seawater's acidity increases, making it more difficult for certain marine organisms to make the shell and skeletal structures on which they rely for survival. This affects plankton, coral reefs, shellfish, and more. And we'll be hearing more about that later. Ocean acidification negatively affects fisheries and the ecosystems that protect our shorelines. NSF investments in ocean observation systems have given us information on the ocean's changing chemistry, including its acidity level. For example, OOI, the Ocean Observatories Initiative, is a science-driven ocean observing network that delivers real-time data from more than 800 instruments. Likewise, the GeoTraces program is an international effort to monitor trace elements and other aspects of ocean chemistry to help monitor climate-related changes in the ocean's chemical composition. Both the structure and circulation of the oceans are affected by climate change. Warmer surface waters inhibit mixing of ocean waters that would circulate nutrients and oxygen, exacerbating deoxygenation or the dead zones. Climate-related change in the ocean circulation can also affect seawater temperatures, nutrient levels, and productivity, resulting in major impacts, including altering weather patterns, changing species distributions, and damaging fisheries. NSF has invested in the facilities, people, making these important discoveries. For example, overturning in the Subpolar North Atlantic Program, or OSNAP, an international collaborative project, is documenting changes in ocean circulation in the North Atlantic and Europe, and its impacts on air temperature, rainfall, and wind strength. Sea level is rising due to the increased melting of land-based ice, which drains into the ocean basins, and the thermal expansion of seawater. NSF is funding the scientists that advance our understanding of these mechanisms that drive sea level rise, and funding the cultivation of solutions to change coastal conditions. For example, NSF's Coastlines and People program, aka COPE, brings scientists, coastal stakeholders, and communities together to jointly develop solutions to mitigate and build resilience against the threats posed by climate change. However, President Biden has also said that despite the shadow of climate change, the future has enormous hope and opportunity. He said that when he thinks of climate change and our responses to it, He thinks of the jobs it will create in renewable energy and also science and technology. Antony Blinken, the US Secretary of State, said that it would be a mistake to see climate only through the prism of threats. As we take concrete actions to reduce emissions and avoid the impacts of climate change, we have an opportunity. One example where NSF has been on the cutting edge is through the recent Convergence Accelerator solicitation on the networked blue economy. This new funding opportunity brings together teams of experts and stakeholders in an innovative way to tackle some of the most pressing ocean challenges of our time, including climate change and its effects. Promoting and facilitating innovative and practical solutions to the complex problems facing the ocean and ocean science will require bringing together teams with diverse backgrounds, skills, and new ideas. This presents an opportunity for scientists to collaborate with the marine stakeholder communities, but also the use of cutting-edge technologies to help address ocean challenges and to aid the sustainable development of the ocean, aka the blue economy. Or as President Biden pointed out, creating solutions to climate change can also lead to new ideas, new technology, new businesses, new livelihoods and careers. Now, let me speak to our community's greatest challenge, creating a just, equitable, diverse, and inclusive community. To deal with the forthcoming challenges in the ocean, we need an innovative, multidisciplinary, diverse, collaborative, and dynamic funding talent pool within the ocean sciences in general. Diverse backgrounds, perspectives, skills, talents, and worldviews lead to diverse ideas and solutions. Here at NSF, we strive to cultivate an education and research ecosystem that is inclusive and collaborative. The ocean science enterprise is strongest when we prepare a diverse, globally engaged STEM workforce. To do this, we are expanding efforts to broaden participation from minoritized groups and diverse institutions in NSF activities across all geographical regions. For example, the NSF Includes program stands for Inclusion Across the Nation of Communities of Learners of Underrepresented Discoverers in Engineering and Science. It is a national program aimed at catalyzing the STEM enterprise to embrace diversity so that the STEM workforce reflects the diversity of the nation. In the ocean sciences, the NSF STEM Student Experiences Aboard Ships, or STEMCs, a program is a 3-year partnership between Indiana University of Pennsylvania, Lamont Doherty Earth Observatory at Columbia, and the University National Oceanographic Laboratory System, or UNOLS, striving for 3 goals: increasing the number of diversity of students pursuing degrees and careers in geosciences. 2, preparing students for all possible career trajectories in the geoscience. And 3, contributing to the evidence base for effective student engagement, learning, and retention in STEM. With the Consortium of Ocean Leadership, NSF has funded workshops to discuss the attrition of women in the academic ocean science leadership pipeline. Other workshops have discussed policies and best practices to address sexual harassment in ocean science field research and how to reach new demographics, such as rural communities, through informal ocean science education. Programs like these are a good start to broadening participation, but we have much more work to do. We must continue to improve the overall safety, inclusivity, and productivity of research settings to attract, empower, and retain the best and the brightest. But there are challenges we urgently need to address. As a recent dramatic example, the pandemic did not affect all scientists equally. Early career scientists and female scientists were particularly hard hit, as were scientists in minority-serving institutions, especially tribal colleges and historically Black colleges and universities. NSF has made efforts to support our scientists through these difficult times. As the pandemic recedes, we also need to look at the lessons we have learned regarding assisting people in the hardest hit demographics. Questions to ask ourselves include: how can we better support scientists who have young children or who are caregivers? How can we better level the playing field for scientists and students with physical or learning disabilities? How can we aid scientists and students who are not born to privilege so that they are not penalized or prevented from achieving their full potential? How can we ensure quality for scientists and STEM students, regardless of their gender, race, upbringing, family status, culture, sexual orientation, age, or geographic location? In a time of grave ocean challenges and ocean science needs, but also a time of increased competitiveness for science positions and funding, how can we ensure that the students and early career scientists of today can have a long and meaningful career where their talents can be fully utilized and their ideas come to fruition? To help early career scientists, I am pleased to announce that the Division of Ocean Sciences has recently relaunched its postdoctoral fellowship program. These fellowships provide opportunities for scientists early in their careers to work within and across traditional disciplinary lines, develop partnerships, and avail themselves of unique research resources, sites, and facilities. The fellowship program is intended to provide beginning investigators of significant potential with experience that will establish them in positions of leadership in the scientific community. The fellows will include participation in a professional development program that emphasizes development of mentoring skills and that coordinates the involvement of fellows in conferences and activities that are focused on increasing the engagement of minoritized groups in STEM. Applicants who are women, veterans, persons with disabilities, and minoritized folks in STEM who have attended community colleges and minority serving institutions are especially encouraged to apply. I hope that this will be one of many new additional initiatives that we can announce to help ocean scientists of the future gain the essential experience and opportunities that they need to start long and productive career. So now it's time for me to move on with today's program. I thank all of you for taking the time out of your busy schedules to join us today and hear from these excellent researchers who are the voices for the next frontiers in ocean science. We are thrilled that they are sharing their stories and their cutting-edge research with us today. I encourage you all to ask questions of our speakers and engage with them about their research, their career paths, and their insights on the future of the oceans. So now it's my great pleasure to introduce our first speaker of the day. Our first speaker will be Dr. Tamara Pico, an incoming assistant professor at UC Santa Cruz in Earth and Planetary Sciences. She was an NSF postdoctoral scholar and a UC President's Postdoc Fellow at Caltech and UC Santa Cruz, and received her PhD from Harvard University in Earth and Planetary Sciences. Dr. Pico's talk will be on insights into ancient ice sheets from the unconventional sea level records. We are thrilled you are joining us today. Take it away, Dr. Pico.
Hey, thank you so much, Terry. Thanks for that introduction. And thank you for having me. And thank you all for being here. Okay, so thanks so much for inviting me. Today, I'm going to outline some limitations in what we know about past ice sheets. And I'm going to show how important understanding solid Earth ice sheet interactions can be for addressing these knowledge gaps and pushing new research frontiers in ice age studies. I'll point out some of the ways that my research aims to address these knowledge gaps, and in particular, I'll discuss the kinds of non-traditional data sets that I think hold a lot of potential in improving our understanding of sea level and ice sheets in the past. Next. So here's a quick outline for this talk. I'm going to start out by explaining what we don't know about ice sheets and sea level in the past. And next, I'll show the ways that understanding interactions between the solid Earth and ice sheets can help bridge these knowledge gaps. And I'll touch on some research projects that I've worked on in these areas. And in the final section, I'll consider the future of our discipline by beginning with the past, focusing on the importance of remembering the history of colonialism in the earth and ocean sciences. Next. So to start out, we can ask why we want to understand ice sheets and sea level in the past. Ice sheet change is the primary driver of sea level change, and understanding ice sheet stability and variability is critical for projecting future sea level rise today. So here I'm showing how sea level has changed over the last 20,000 years, which is the last time that ice sheets were at their maximum extent. So the y-axis here is sea level, and the x-axis is time, where to the left is in the past. So at the last glacial maximum, sea level was about 130 meters lower than today on average. This image on the left shows cover of continental ice sheets at the last glacial maximum. So all of Canada was covered by ice sheets. And these images towards the right show the cover of ice sheets at present day at today's sea level, which is at 0. These blue lines moving towards the future show projections of how sea level could change in the future under different scenarios of CO2 forcing. So there are large uncertainties in predicting how ice sheets will change in response to climate change. And one major uncertainty is how the solid Earth interacts with ice sheets. For example, errors in estimates of recent Antarctic mass balance based on satellite gravity measurements are actually dominated by the uncertainty in the rate of regional crustal uplift associated with ongoing glacial isostatic adjustment. So today we want to know ongoing ice melt rates, but we also want to know how variable these melt rates can be, how stable are ice sheets, and how much ice is going to melt as we continue to warm the planet. Next. So we can look to the past to try to answer this question. This figure compares CO2 temperature and sea level of present day to past warm periods that might serve as analogs for our future. So the blue bars on the bottom show sea level, and light blue shows uncertainty on past sea level. So for example, at the last interglacial, when CO2 and temperature were similar to pre-industrial times, sea level is thought to be 6 to 9 meters higher than today. And these pink pie charts within that blue bar show the fraction of ice from Greenland and Antarctica that you would have to melt in order to fill the oceans with so much meltwater to raise sea level by 6 to 9 meters. So we can see that as we move backwards in time, so it's moving towards the right, uncertainty on past sea level increases. And to me, that really highlights the need to better understand ice sheets in the past so that we can understand where we're headed today as temperatures and CO2 levels rise. Next. So what kind of data, sorry, go back. So what kind of data are used to consider ice sheets in the past, and what are the gaps in this information? We can map this out on a temporal scale, which I'm plotting on the y-axis. So for example, the knowledge we have about ice sheets at a single given time frame versus over tens of thousands of years. We can also consider the knowledge we have about ice sheets on a spatial scale, which I'm plotting on the x-axis. So for example, at a single location or across a continent or even over the entire globe. Next. So we can go to where the ice sheet was and measure the margin and try to date it. So this gives us information about the border of the ice sheet at certain times, but it can't really tell us about ice thickness and therefore ice volumes. Next. From a sea level perspective, we can use observations of past shorelines from places close to the equator to try to infer past global sea level. So the advantage of this approach is this gives us a really good idea of total ice volume globally, or globally average sea level, but the con is that we know nothing about where and how big each ice sheet was. Next. On the modern timescale, we can use GPS or gravity data to track ice sheet and sea level changes, but this only gives us information about the last few hundred years, not over tens of thousands of years, which is really the glacial scale. Next. To get at this time scale, we can rely on dynamic ice sheet modeling, which uses physics to predict how ice sheets change on this longer time scale. But this often is not well constrained by data, especially during time periods in the past. Next. For example, it's really challenging to reconstruct ice sheet dynamics in the period before the last glacial maximum because there are few sea level records or records of past ice sheet extent. Since ice sheets are somewhat erosive in nature as they grow. And understanding this history ultimately tells us about the pace of ice sheet growth. Does it happen slowly or suddenly? How stable are ice sheets in response to climate variability? So what I hope these examples highlight is that it's challenging to connect the regional to the global scale for ice sheets on the glacial timescale. And I propose looking for unconventional sea level records to approach these questions from new angles. Next. So ice sheets interact with all components of the Earth system, and in order to take advantage of different unconventional sea level data sets, I'll focus on the interaction of ice sheets with the solid Earth in a process called glacial isostatic adjustment. Next. So ice sheets change sea level globally through interactions with the solid Earth. And one way that ice sheets change sea level is they're so massive, they exert a gravitational attraction on surrounding oceans, causing sea level to be higher in their vicinity. Next. So for example, here I'm showing the sea level fingerprint that's caused by melting ice in Greenland from 2003 to 2016. And you can see this blue region around Greenland is an area where sea level actually falls close to a melting ice sheet. Next. And so here's another example of this. So here I'm showing how sea level changes in different parts of the world depending on regional ice melt in Greenland. So the colors in each region of Greenland show how sea level would change locally at 3 different sites. So Aberdeen, Scotland, Boston, USA, and Cape Town, South Africa. So if ice in a certain region were to if ice in a certain region of Greenland were to melt, and the dashed line is the 0 line, warmer colors, red and yellow, are a sea level fall, and cooler colors, green and blue, are a sea level rise. So we can see that in Scotland, sea level will fall if the eastern portion of Greenland melts. In contrast, in Boston, sea level will only fall if the southern sector melts. Otherwise, Boston experiences a smaller than average sea level rise. And in Cape Town, regardless of the region of ice melt in Greenland, sea level will rise. So this is a bit counterintuitive that melting an ice sheet can cause a sea level fall. And that reflects the complex sea level physics that results in geographically variable sea level change. So the world's oceans are not bathtubs that rise and fall uniformly. Local sea level can be quite different from the global average. Next. So in addition, ice sheets load the Earth's crust, displacing mantle from underneath and causing this region to become subsided under its load, causing crustal deformation. Next. Here I show present day sea level change due to this process of glacial isostatic adjustment, where warmer colors are a sea level fall and cooler colors are a sea level rise. And on the right, I'm showing a schematic of this physics. So as the ice sheet grows, it loads the crust, pushing mantle out and displacing it from underneath the ice sheet, forcing that mantle to flow outwards to the periphery of the ice sheet, which then gets uplifted in what's termed the peripheral bulge. And as the ice sheet melts, the crust is unloaded and mantle can flow back beneath the former ice sheet as the crust rebounds. So we can see that most of Canada is that's in red here is a region where sea level is now falling. Next. And this is evidenced by uplifted shorelines such as these I'm showing here on the left in Arctic Canada. You can see the series of past shorelines that have now been uplifted as a result of post-glacial rebound. Next. So understanding solid Earth ice sheet interactions, such as glacial isostatic adjustment, has the potential to bridge current knowledge gaps in ice age research. And I want to point out what I see as the frontiers for learning about past ice sheets and past sea level variability. Next. So in my research, my goal is to find data that bridges these gaps, and I use glacial isostatic adjustment as a tool for taking advantage of unconventional data sets to learn more about past ice sheets. So for example, I showed that we can gain additional information about regional ice sheets by using sea level records near former ice sheets to gain an idea of a longer term ice sheet history. Next. So traditionally, people use sea level records that are far from ice sheets that are near the equator to try to avoid contaminating a sea level signal with glacial isostatic adjustment. But I've suggested kind of taking the opposite approach and actually go close to where the ice sheet used to be and find sea level records that are peripheral to the ice sheet that have a really large glacial isostatic adjustment signal in order to allow new insight into regional ice sheet history. So on the right, I'm showing again the schematic of how a peripheral bulge grows. So as the ice sheet over Canada grows, it loads the crust, forcing mantle from beneath to flow outward to the periphery of the ice sheet in this region termed the peripheral bulge, which then gets uplifted. And if we look at this map on the left, we can see there's this blue ring around the North American ice sheets, and that covers all of the United States. And that's the region that gets uplifted as the ice sheet grows. And we can see that the US East Coast, so where NSF headquarters are, so it's kind of right in the middle of that peripheral bulge. And that means that as that sea level history in Washington, DC, for example, is really sensitive to the ice sheet history over Canada. So as the ice sheet grows and melts, this land is going up and down. And so that sea level history in part reflects the history of ice loading nearby. Next. So in the study, we used anomalously high sea level markers on the US East Coast to infer the longer term loading history of the North American ice sheets. And we found that to fit these sea level records, a late growth of the eastern part of North American ice sheets is required starting around 45,000 years ago. And this suggests that North American ice sheets are the main contributor to a global sea level fall, to a rapid global sea level fall, leading into the last glacial maximum. So this top figure shows a revision to thoughts about reconstructed global ice volume histories. So whereas past sea level reconstructions that are based on oxygen isotopes suggest a slow and steady buildup of ice leading to the last glacial maximum, this is kind of a canonical idea of how ice sheets grow. My work suggests ice volumes varied substantially, and there was a really rapid growth of ice in the period leading into the last glacial maximum, and that much of that sea level fall can be attributed to a really rapid growth of North American ice sheets. Next. I've also shown how other alternate data sets can be used as a kind of sea level indicator. For example, using the history of landscapes near ice sheets. So rivers are really sensitive to slope, and we can explore how landscapes respond to slope changes due to crustal deformation on the Ice Age timescale. Next. So a late and rapid growth of North American ice sheets would induce uplift of the peripheral bulge over the United States, tilting topographies and causing landscapes to respond to changes in slope. In a couple of papers on US East Coast rivers, I showed that glacial isostatic adjustment may be responsible for changes in river dynamics that were coincident with the timing of peripheral bulge uplift during MIS3, this period leading into the last glacial maximum. Next. So these studies suggested that ice sheets might have grown rapidly in the period leading to the last glacial maximum, in contrast to slow and steady growth of ice sheets across the glaciation phase, as inferred in oxygen isotope records, such as the one shown here from Wasiki and Rainbow. Next. So another limitation in our understanding of ice sheet stability is knowing when, where, and how much ice melted as the ice sheets retreated over the last deglaciation. For example, we don't understand how much ice melted from the region between the two North American ice sheets. So on the right, you can see there's the Cordilleran to the left and the Laurentide to the right. We have some information about ice margins as these ice sheets retreated, but we don't know very much about ice thickness change, and knowing the volume of ice melt is really important for thinking about how meltwater has influenced oceans and global climate. Next. So I've shown that we can use the history of ocean connections nearby ice sheets to improve our understanding of ice sheet stability over this time period. Next. So in one study, we used the flooding history of the Bering Strait, which connects the Arctic and Pacific Oceans during the last deglaciation, or the last melting time the ice sheets were melting, to infer the timing of melt of the ice saddle that connected these two North American ice sheets, the Cordilleran towards the Pacific and the Laurentide towards the west. So the Bering Strait sea level is really sensitive to nearby ice sheet melting, as it's close enough that that gravitational impact of ice sheets on the ocean, which I talked about earlier in this talk, that gravitational impact becomes a really important control on sea level. And we found that to explain the observed two-phase flooding history during the last deglaciation at the Bering Strait, a substantial melt of the saddle region, so the region of ice that connects these two North American ice sheets, would have had to occur from 13 to 11.5 thousand years ago during the Younger Dryas cold period. Next. In other work, I showed that we can even use past ice stream dynamics to gain additional information about the history of ancient ice sheets. Next. So this was a follow-up study to that Bering Strait research to understand the melting history of the North American ice sheet saddle. So this region of ice that connects the two North American ice sheets, the Cordilleran to the Laurentide. So evidence of ice stream retreat can differentiate between different possible ice loading histories because the history of ice sheet loading changes topography through glacial isostatic adjustment. So crustal deformation from the weight of ice sheets, either as they load or unload the crust, changes the slope beneath ice streams. And ice streams are very sensitive to underlying slope, especially on a retrograde slope or a reverse bedded slope. And this can produce what's known as a marine ice sheet instability. So in this study, we found that the saddle, this area of ice that connects the two North American ice sheets, melting during the Younger Dryas from 13 to 11.5 thousand years ago is most consistent with geologic evidence for a very rapid collapse of the Amundsen Gulf ice stream at the start of the Younger Dryas that's been attributed to a marine ice sheet instability. And I think what is really interesting about this study is it shows that we can use short-lived changes in a local ice stream behavior to reveal much larger scale and longer term ice sheet dynamics. So if the stability of an ice stream is dependent on slope, and that slope is ultimately controlled by kind of continental scale load of the ice sheet, we can use evidence for the dynamics of that ice stream to tell us about a much bigger and longer term history of past ice sheets. Next. So ultimately, each of these examples highlights how understanding solid Earth ice sheet interactions can help us to leverage new insights from data sets that aren't traditionally used to learn about past ice sheets or sea level. Next. So I'm going to move on to the last section of my talk, which focuses on the theme for the symposium, futures. And rather than focus solely on futures, I'll suggest we work to connect past to future. I've become quite interested in thinking about cultures of exclusion in the geosciences. And I got involved in research on these topics through a PhD secondary in feminist studies. And here I'm just showing a few of the projects that I've worked on in this area. And today I want to spend a few minutes discussing a curriculum project that I've been leading called Geocontext. Next. We've been creating a collection of teaching modules that provide a social and political context of both historical and modern practices in geoscience. So we carry traces of the past with us through cultures and practices, especially when we revere certain parts of history without considering other sides. My hope is that incorporating these parts of our history as a discipline that have been excluded as irrelevant will help to train the next generation of geoscientists to explicitly detach themselves from cultures and practices of oppression and exclusivity. So to me, these histories serve as a kind of counter storytelling, reframing the scientific discovery of geologic principles as an outcome of a colonialist project, which was aimed at maintaining white supremacy. These stories provide another angle to the unilateral training we've often received in the geosciences, either implicitly or explicitly, about the social neutrality of science and the heroism of individuals that are considered the founders of our discipline. Next. So to give you a taste for these modules, I'll describe some of the content included in the oceanography module, which was created by Wesley Wiggins during an undergraduate summer internship. And I encourage you to check out the full module later. So modern oceanography developed this discipline in the 19th century with ocean depth sounding and charting of surface currents. And understanding surface currents and trade winds is really motivated by improving commercial trade ship navigation. And at this time, a lot of commercial trade ship use was related to the slave trade. So between 1810 and 1860, ships transported 3.5 million enslaved people across the Atlantic. And this figure on the left kind of shows the coincidence of ship trade routes and northeast trade winds. On the right, I'm showing a portrait of Matthew Fontaine Maury, who was a key early oceanographer who published the first modern oceanography textbook in 1855. And this picture here shows a trade wind chart that he created. So Maury was in the US Navy, and he was also a professor at the Virginia Military Institute, as well as having served in the Confederate Navy during the Civil War. So Maury had some ideas that were kind of both related to continuation of US slavery and his work in oceanography. He envisioned continuation of US slavery in Brazil as the Amazonian Republic, is what he termed this. And this wasn't just Maury, there were other people at the time that were arguing for continued US slave holding in Brazil. He proposed that US slaveholders would own property in Brazil, which he considered, like the US, to be a slave country, based on the similar climate and geography of the Amazon to the Mississippi. So like other scientists of his time, Maury believed that certain climates or topographies were well suited for the institution of slavery. And if you want to learn more about this, you could also check out the landscapes module in geocontext, which talks more about the scientific racism, connecting race and geography. So based on the pattern of ocean surface currents, Maury argued that Brazil was a natural extension of the US empire, as surface currents would hasten travel between the southern US and Brazil and the Caribbean. Maury envisioned a continuation of agricultural production by US-owned slaves, and the plausibility of this Amazonian Republic, as he called it, was actually tested by the US Navy by exploring the navigability of the Amazon River at Maury's suggestion. And to me, more than Maury's Confederate legacy, I'm really fascinated by the ways that politics and science are entwined in Maury's research and kind of that legacy today. Next slide. But ultimately, I'm really interested in considering the legacy of 19th century US geologists' connection to science that was motivated by colonialism and scientific racism, particularly in how traces of this past emerges in cultures, training, and practices today. For example, we can't ignore the history of the outdoors as a site of exclusion or violence for people of color, as it feeds into the perceived and actual safety of the field for undergraduate students or practicing earth scientists. Early US geologists made claims about what kind of bodies should inhabit what kinds of spaces or land and for what purpose, using geography and climate as justification. So the safety of undergraduates today in the field is not divorced from the legacy of early scientists in our own discipline. Early US geologists were entrenched in scientific racism, using the study of the earth and the oceans to further colonial objectives. I argue that ignoring this history, which is seldom known or mentioned in undergraduate courses, leaves an absence in knowledge that would help students contextualize their own experience. In a discipline where underrepresented minorities make up less than 7% of undergraduate geoscience majors, students of color are not likely to have their experiences validated by others, and maybe even less likely to have their testimony successfully transmitted to an instructor. So by sharing this knowledge, I hope students can be empowered to understand the issues that they experience as they navigate discipline, such as cultures of whiteness, masculinity, and exploitation. We can recognize certain racist ideas as the legacy of scientists that came before us, which I think will make us better equipped to critically consider our own legacies and the impact that we make. Next. Oh, I think it's missing a slide. Go back. It's missing a slide. I'll just end. So in contrast to scientific objectivity, that's assumed to be value neutral, a strong objectivity requires us to understand the practice of science in a social context, where the political, economic, and social values of the scientific community control the entire scientific process. I'll end here. Encourage you to learn more about this picture here. Sorry, that other slide is missing, but this is a picture from the interview about philosophy and we're interested. So with that, I'll just summarize. At the beginning of this talk, I kind of focused on what we don't know about ice sheets in the past and sea level in the past. And then I showed how we can use understanding of ice sheet solid earth interactions to gain more insight into past ice sheets by using non-traditional sea level data sets. And finally, I showed a bit about how remembering histories of racism and colonialism in our discipline can help us build towards the future. So next slide. So with that, I just want to acknowledge my collaborators who have just been really fantastic to work with, and funding and support that I've gotten much from NSF. So thank you. And I'll end there and can end. The next slide is a summary slide we can end with. Okay. Thanks.
Well, that was terrific, Tamara. Very powerful. And what an absolute way to start the symposium for the day. Back in the day, I actually did spend some research time studying sea level. And so that to me was like a back to the future thing. It was great. So thank you for that. So let me ask you this sort of question related to the first part of your talk in terms of sea level. I think you took an interesting approach, right? So you sought to identify gaps in the understanding of sea level history using unconventional data sources. I thought that was really intriguing. So when you did that, what surprises did you get from using that? And did you find anything that was just completely unexpected? And how did you work relative to like the previous hypothesis of sea level for that? So kind of a three-parter, and I'm happy for you to take it on any way you want.
Yeah, I think most of these research projects, the results were unexpected, and that's probably something we've all dealt with as maybe the most exciting part, right? When it comes out not what we expected. So I guess I would say a lot of it was not what I expected going in, and I guess I'll talk specifically about the Bering Strait study. Going in, actually, originally, I thought that this sea level record, it's not exactly a sea level record, right? But the flooding history of the Bering Strait would help us understand how ice sheets melted during meltwater pulse 1A, so around 14.5 thousand years ago. And most people have suggested that the ice saddle between these two North American ice sheets collapsed during meltwater pulse 1A, which is a time when global sea level rose 15 to 20 meters in just 500 years. And so it kind of makes sense that the ice saddle holds a lot of ice volume. If that melted at that time, that could explain a lot of that sea level rise. But what we found, I was kind of playing around with all these different scenarios and kind of like, oh, there's got to be a way to, match up the sea level record to the ice melting at this time. And it just, that wasn't working. And instead, it looked like the ice saddle was melting later. So that was kind of surprising result. But that's been interesting to then kind of say, well, that's a surprising result. How else can we test it? Which was kind of where that idea of can we use the ice stream as this other constraint in this time and area, and like geographically and in time, we just don't have a lot of data for what's happening in this region of the ice sheets. So that is, yeah, all of these projects have been fun in that way, that they've all kind of been surprising. Yeah.
That's awesome. Yeah, it was just really like that whole approach. Sometimes people thought we'll go start someplace maybe like in the equatorial regions because sort of the GIA is small there, but actually if you go to proximal sites, GIA is large, but if you can model it correctly, you can get a very solid answer in situ, right?
Yeah, absolutely. That, you can use the disadvantage as the advantage to learn about something else. Yeah.
Yeah. That was awesome. I was like, ooh, okay. And then you ended at sort of on the second part of the talk about sort of going into sort of colonial science versus sort of indigenous knowledge kind of areas. And I thought that was also very strong. And there's a lot of work being done in these areas. And so I guess, how can we sort of talk about those as we move forward? Because you are correct in the comment how we grew up, somebody in my age group grew up learning about all the people you talked about, but without, in a colonial sense, not an indigenous knowledge sense. Can you talk a little bit about how we can confront those sort of two convergent, the colonial science versus what actually indigenous people and others were actually doing at that time?
Yeah, well, so I'll start by saying, so it took us a long time to come up with the name, but when we called it geocontext, it felt like that was just right, because what we want is providing that context to, like what you said, how we grew up in the sciences hearing one story without a context for what was happening in that story. What were the other angles to that story? What else was going on? And so I think that, without being redundant, I think that's really important, right? To recognize that every story, there's so many different ways to tell that story. And the more information we have around it, the more context we have, the more we can understand what was motivating it, what were the values at that time, and what part of that is left still in our discipline. And so I didn't really talk about indigenous knowledge systems today, and actually geocontext kind of touches, some of the modules touch on indigenous knowledge a little bit, but I would say, I think, recognizing, I think it's really important to see science as bigger than the way we've been trained as seeing science, right? And that allows us to acknowledge different forms of knowledge being science. So I guess related to indigenous sciences. And what was I about to say? Oh, that, so I guess I would say, I think there's, geocontext doesn't fully talk about indigenous sciences, but what I will say related to kind of this narrative, what we can do to maybe ask questions of the narrative we've been told, or to fill in gaps about the narrative that we've grown up hearing, is thinking about what we haven't heard. What are the histories written by someone, right? Like someone tells what history is, and we actually aren't going to know all the stories that were happening at that time that weren't told by the people that we're listening to. And so that's where this kind of, I hope that geocontext kind of as a counter storytelling can inspire other people to do the same, to ask what are the other stories that might be possible? And it might not be something that we can actually uncover and point to and have an archive of, because those weren't the stories that were being recorded by the people at this time, right? That were like, we know who they were. They were, the people that were either big in the sciences, or even just kind of making decisions at a policy level in the US or other kind of, imperial countries. And so I guess what I would say is that we can use our imagination, because we might not have those stories in the archive, but we can actually fill in the archive by imagining what would be the counter story, what was happening at the time, what were, as certain geologists are going into these space, they're interacting with other people, and maybe we didn't record what those other people were thinking about what they were doing, but we can use our imagination to kind of try to fill in the archive and use that as a resistance to the narrative that we've been told.
No, that's terrific. And I love the name geocontext, right? Because it's the classic play on the words of place things in context, right? So that was awesome. So I've got homework to do now. And that's always a good way to start a symposium. So thank you, Dr. Pico, for a wonderful thought presentation and some things that we all have homework to do after this. So that was great. Thank you.
Hey, thank you so much, Terry.
You're very welcome. So, Chris, we go on.
Yes, we're just switching over the slide deck. So I think Elena and Angel are ready to share Jacqueline's slides coming up here.
And there was a sort of a note for Dr. Pico about notion about potentially sharing your syllabi if possible. They sound incredible, and I would agree with that. I'm like thinking to myself, when I returned from my position here at NSF back to UT Austin, I've got to very much update my my intro oceanography slides. So, all right.
We're getting through the slide quality looks great. So I think we've fixed that problem. So everything is A-OK, and we're exactly on time. So
look at that. I'll try to help with that. How's that great? So our next speaker is Dr. Jacqueline Padilla-Gamiño, and she's an assistant professor in the School of Aquatic and Fishery Sciences at the University of Washington, UW. Dr. Padilla-Gamiño earned her bachelor's degree in oceanography at the Autonomous University of Baja California in Mexico. She won a Fulbright scholarship to do a master's degree in biology from Cal State and gained a PhD in oceanography from the University of Hawaii at Manoa. Dr. Padilla-Gamiño's talk today is entitled, there it is on the screen, The Significance of Life History Strategies in Coral Recovery After a Bleaching Event. The screen is yours. Take over.
Thank you, Terry. And
you're welcome.
Thank you so much for the opportunity to share my work. Right now, do you see me? Because I don't see my face. Yes? Okay, great. Perfect. So yes, today I'm going to talk about the significance of life history strategies in coral recovery after a bleaching event. Next, please. So the fitness of an organism is not only going to depend on the ability to survive, but also on the ability of reproduce. So because of this, nature is going to invest heavily in both the art of survival and the art of reproduction. Next, please. And sexual reproduction is very important because it's going to promote genetic variation and increase the potential for adaptation, which is critical in the context of a changing climate. Right? Having sexual reproduction is really important to generate new mixes of genes that can potentially lead to more adaptations to deal with the environment. Next. But sexual reproduction is a complex process, right, that requires multiple steps, such as sex determination, gametogenesis, the timing when the gametes are released, how the mating behavior, fertilization, and brooding, right? And all of these steps can potentially be bottlenecks depending on the biological and physical conditions of the environment. Next. And there are still many mechanisms associated with sexual reproduction that we don't really understand, that are understudied. And some of them are highlighted here. Next. And they include gametogenesis and allocation, spawning and gamete quality, and fertilization. And within these categories, you can see that I have other subcategories, just kind of to show the processes that are associated with the big categories. And I will say that we don't know much about them, especially in the context of what's happening in the field, right? How much they reproduce, how often, and when. And today, I'm going to talk to you about specifically the blue category, and specifically about provisioning and gametogenesis in corals. Next. So corals create coral reefs. And although they do not occupy a very large area, they host incredible amount of biodiversity. And this is because these are like beautiful tropical underwater cities where organisms go to breathe, to live, to feed, to reproduce. And so they are extremely important for the ecosystem, but also for humans. Next, and human activities. And the coral reefs growth and health depends a lot on a symbiosis between the corals and dinoflagellate endosymbionts. So the dinoflagellate endosymbionts are gonna live within the tissue of the corals, photosynthesize, and provide glucose and oxygen, which are products of photosynthesis that the coral can use. And the coral will provide nutrients and protection to the algal symbionts. And this relationship is very critical, very effective, and very important to have these massive reefs that we can find in tropical seas. Next. But the problem when this relationship between symbionts and polyp, the problem is that it can break. This relationship can be susceptible, and corals can lose their symbionts due to stresses such as thermal stress, but they can also lose it because pollution and or chemicals. And so unfortunately, recently, a lot of corals have been losing their symbionts, and this is called bleaching. And they have been losing them as a response to thermal stress. So rising seawater temperatures have been causing coral bleaching at a massive scale in the last decades. And as a result of these bleaching events and other anthropogenic activities, over 50% of the world's coral reefs have died in the last years. And lately, we have found that these bleaching events are happening more often and for longer periods of time. And as you can imagine, if we lose the corals, it's like we're losing the trees of the forest, right? We lose biocomplexity, and that has impacts for the species living on them or depending on them, like many coastal communities around the world. Next, please. And so different corals are going to have different bleaching thresholds depending on where they are located, depending on the species. And so for Hawaii, and here I'm showing you at the top picture is a nice, healthy coral colony of Montipora capitata, and at the bottom I'm showing a bleached colony, right? They look bleached or white because they have lost the color that the symbionts were providing to them. And so in the right graph, what I have is on the x-axis, I have month, on the y-axis, I have temperature. And the gray line represents kind of the mean temperature between 1994 and 2013. And the orange lines represent the temperature that we have in 2014 when we had a major bleaching event. Next. And so when temperature hits 28, that's when corals in Hawaii generally start bleaching. Next. But when temperatures hit 30 degrees, that's when corals start to die because of bleaching. Right, when corals are bleached, thank you. And when corals bleach, they tend to, it's not necessarily that they die, they become very vulnerable, right? And they are not receiving all this energy provided by the symbiont. And if the stress reduces or is reduced, then corals can regain symbionts and recover. But if the stress lasts for too long and they are vulnerable, then they might die. Right, so that's the things that happen. And so we saw that in 2014, we saw it in 2015 again, and you can see how in 2015, actually the thermal stress occurs more intense, right, higher temperatures and for longer time. And these bleaching events are just becoming more and more frequent. And 2014 and 2015 have been the worst bleaching years in Hawaii. Next, please. And so before I get to my research questions, I just want to give a little bit of background on coral reproduction and the coral life cycle. Right, so corals, when they reproduce, they release the gametes, egg and sperm, into the water column for external fertilization. And those are called spawners. Next, the sperm will fertilize the egg, and then the embryo will develop, next, and develop as a larvae or planulae. And so this happens in the water column, next. But other corals can be brooders, and for brooders, the internal fertilization occurs inside the coral mom or the female, and the larva is released. Next, but eventually, regardless if the planula came from a brooder or a spawner, this larva will settle down, metamorphose, and then become a polyp, next, that will continue to grow until forming another coral colony. And so in this talk, I will talk to you about two coral species that are spawners. One of them is hermaphrodite, the other one is gonochoric, meaning that it's either a male colony or female colony. Next. And so bleaching, there are several studies that have documented the consequences of bleaching in reproduction. And some of these effects have been reduction in egg size, fecundity and spawning, total reproductive failure for several seasons, maybe a premature release of the gametes or resorption of the gametes, where gametes are just resorbed and they are not even released. Next. However, Hawaiian corals can still reproduce despite bleaching. And this was an observation that Fanny Cox did in 2007, when there was a not so strong bleaching event. And so the picture that I'm showing you here is the area where I study, Kaneohe Bay, where you can see the corals that I'm gonna be talking about. So the ones that look orangish is Montipora capitata, and the one that looks yellowish is gonna be Porites compressa. Next. And so those observations from Fanny Cox and my observations as well, when I look at the coral that was reproductive, even though it has gone through a massive, right, thermal stress, is like, how can they do it, right? Like, and what is the variability in response to stress in the context of reproduction, right? So the first question that I'm going to address and talk to you about is learn about the reproductive ecology and physiology after bleaching. And this is really important because this question is super associated with the recovery phase of corals, right? We know already a lot what happens when a coral is bleached. We know that there's a lot of, oxidative stress and heat shock proteins, right? But what happens during recovery? Which is a phase that is just as important, if not more important. Are they going to be able to reproduce? Do they acquire the same physiological signature post-bleaching? And how long does it take to have this regular physiological signature? The second question is about where is the energy coming from to produce the eggs, right? So if the corals are bleached, how do they acquire energy so that the eggs continue to develop? And finally, are there any trade-offs in the resource allocation following a bleaching event in corals that have very different life history strategies, like being hermaphrodite or being a gonochoric species? Next. So I will start with the first one, and yes, which is like the reproductive ecology and physiology after bleaching. And for this, next, So for these questions and for this project, I work in Kaneohe Bay, which is located in the windward side of Oahu, like you can see in the little map on the left. Next. And so in some areas of Kaneohe Bay, you can really see this type of view, right? You can see that in some areas, Montipora capitata and Porairis compressa covered 100% the benthos. Those are, you know, it looks extremely beautiful. Again, Montipora capitata is the orange morphotype or color, and the more yellowish is Porairis compressa. They grow at similar rates, so they're often competing for space. Next. And you can see that very clearly in this picture, where you can see how the entire space is occupied, and you can see the different corals pushing, right, to gain some space. And as you can see here, that there is a lot of phenotypic plasticity. So especially in both corals, but especially Montipora capitata, that shows extreme phenotypic plasticity. Sometimes it can look more like a plate, but sometimes more like fingers, and anything in between. Next. And so Montipora capitata is also known as a rice coral. It's a main reef builder in Hawaii. This is a simultaneous hermaphrodite, meaning that each polyp can produce egg and sperm, and it has synchronized event to release these egg sperm bundles to the water column, like you can see on the picture on the top right. Right, these egg sperm bundles are kind of pinkish and float very slowly all the way to the surface and to the top because of the high lipid content in the eggs. And when you are there snorkeling or diving, it's almost like seeing snow, but it's upside down and it's pink. And it's going backwards, right? Like it's going backwards and it's pink. And in the bottom, in the picture at the bottom, what you see is an egg sperm bundle, a close-up. In the center, you see the sperm, and surrounding the sperm package are all the eggs. This bundle eventually will disintegrate, the eggs will hydrate, and then the eggs will be ready for fertilization. And this coral specifically releases large quantities of egg sperm bundles compared to Porairis compressa. Next. So Porairis compressa is different in the sense that it's a gonochoric species. So they are gonna release a lot of eggs and sperm. So, but some colonies will be males, other colonies will be female. The spawning events, interestingly, are not as synchronized as the ones for Montipora capitata. And overall, Porairis compressa tends to have lower fecundity, but the cellulars have higher growth and survival compared to Montipora capitata. And I'm showing these characteristics just to kind of show the different life history traits that each of these species has. And because we're gonna look into all these different things, the reproductive ecology, the physiology, where is the energy going, and if there are any trade-offs in allocation after these two species are going to bleach. Next, thanks. And so here I'm showing you what I think is a very cool graph, because so on the x-axis is our month, and I have 3 y-axes. Temperature on the left side, which is represented by this blue line. And you can see how temperature increases throughout the year. And then you have solar radiation, which is the brown line. And then you see egg size, which is represented by the bar graphs. And the first main thing that you can see is that eggs from Montipora capitata, and this graph will be very similar for Porairis compressa because they have a very similar gametogenic cycle. The eggs start forming around October, right? And it takes all the way to June before they are ready to be released. So just developing gametes is a process that takes between 7 or 9 months for some corals. So it's a very long process, and this is just egg development. Next, spawning or the release of gametes occurs between June and September for both species. Next, but what is really important to notice is that, next, please, is that bleaching actually occurs between September, October, and November, in a period where the eggs are developing, right? And so it's really important to keep in mind not just the bleaching window, but what's going to happen during the recovery before the spawning season when the eggs are actually developing, right? And as bleaching continues to increase in frequency and duration, this bleaching window might expand and might have consequences for the gametogenic cycle and the synchronization of the spawning and the development of the gametes. Next. So we did a study looking at, what we did is we collect colonies, or I guess during a bleaching period, what we did is we went to different sides of the bay to tag colonies that either bleach or did not bleach. Because in terms of bleaching, there is a lot of variations. Some colonies will die, some colonies will bleach and recover, and some do not bleach at all. And so we tag colonies that bleached and colonies that did not bleach at these sites for the two species. And we had two patch reefs and two fringing reefs. Patch reefs generally receive higher water flow. They are cooler compared to fringing reefs that are also reefs where receive sedimentation and riverine input. Next. And so this is a graph showing the percent corals that were reproductive for the two different groups, right? So what we did is after we tagged them, we left those colonies and we monitored them as they recovered. And 7 months later, we went there to obtain samples to see whether they had any eggs in them or egg and sperm on them. And so what I'm showing here is the percent of corals that had gametes for the 4 different sites. And to our surprise, we were really not expecting this. Montipora capitata colonies that bleached actually had a higher likelihood of having eggs compared to the ones that did not bleach. And this is not really what we were expecting. We knew that bleached corals could develop gametes, but we never thought that the likelihood of finding gametes in these colonies will be higher compared to the ones that did not bleach and remain what we think healthy. Next. For Poraitis compressa, it was a different pattern. The one that we expected for the most part. So in 3 of the 4 sites, we found that the non-bleached colonies actually had a higher percentage of corals that were reproductive with respect to the bleach category that show lower percentage of corals being reproductive, except for patch 1, which had the similar percentage of corals that were reproductive. So Montipora capitata has higher percentage of bleach individuals with eggs, whereas Poraitis compressa had lower percent of bleach individuals with eggs. Next. Another really interesting thing that we noticed with Poraitis compressa, which is gonochoric, is we were able to separate the sexes. And we overall found that in 3 out of 4 sites, the male colonies are more likely to bleach compared to the female colonies. And why this is happening is part of my next story, but I don't think this story is complete yet, and it's quite amazing and interesting to know that possibly a lot of the bleaching variation that we see in the field might be due to the fact of just having different sexes, right? But most of the time, because they're really hard to tell apart, you need histological slides to really confirm that they have egg or sperm. Most of the times, coral ecophysiologists perform these studies without knowing that they are male or female, and this might be not leading us to really understand the mechanisms and the real responses of the populations. Next. And so what we did next is, in addition to get samples to see whether they were reproductive or not, we took samples to look at the physiology via transcriptomics. So we look at the gene expression of these bleached and non-bleached corals at different sites, and we only did it for Reef 44, which is a patch Reef 1 at the northern part of the bay, and fringing Reef 2. And what you can see for both sides, and on the left side, I have a plot of a discriminant analysis of principal components showing the physiological signatures between these two groups of corals for the two different sites. And you can see that for both sides, there was a very distinct physiological signature between bleached and non-bleached corals. But let me remind you that these corals is the physiological signature after 7 months from the bleaching event, showing the lasting impacts of bleaching on gene expression. So looking at just gene expression, we could tell apart which one had bleached and which one had not. And these distinctions were more evident in the fringing reef site compared to the patch reefs. So you can see how the purple centroid is farther away from the yellow centroid of fringing reef 2, right, between the bleach and the non-bleach categories. And for us, it was surprising, right, the legacy of stress, the legacy that bleaching has on the physiological signatures of this species. Next. And this graph is just for the females Porites compressa, right? Because we did it knowing that we know that males and females are behaving differently. Next. And in this slide, I can show a little bit of the gene expression so that you can see how bleach females and bleach males actually had a very distinct gene expression patterns. And overall, on the right graph, I'm showing the upregulated differentially expressed genes. Overall, the males tend to have more of the regulated differentially expressed genes with respect to females. Kind of aligning with what we found, that the males were more likely to bleach, and perhaps the males are just having more upregulated genes, are perhaps more physiologically active, trying to deal with getting better, recovering, to be ready for the spawning season. Next. And these are just some of the biological functions that were also very different between females and males, showing transport processes, cell division processes. A very interesting one was mitochondrial and membrane organization, differences between female and male, most likely associated with the transmission of symbionts to the eggs. And a very important one, very more percent terms in immune system processes for the females. Perhaps because of this, females might be less likely to bleach with respect to males. But I think there are still a lot of unknowns in that story. Next. So that was my first question. And then the second question is, okay, so for the ones at the colonies that bleach, where are they getting the energy to produce the eggs? Next. So if you don't have any algal symbionts, what can the corals do to obtain energy? Next. And one big answer, and there is a lot of work on this, is they can feed, right? And they can probably get energy. But so far, there has not been a study that has shown evidence that heterotrophic nutrition can actually provide carbon to the eggs. There's only one study that has shown that symbionts provide carbon to the larvae, but in the context of spawners, we don't know anything in terms of autotrophy, and there's nothing on heterotrophy nutrition participating in egg development. So that's the question. Next. And so we have a few hypotheses. And so here I'm just showing you the coral here, just kind of to orient you. You have the poly with the tentacles, and then you have the gastrovascular cavity or stomach next. And so this is gonna be where the eggs develop along the mesenteries. Next. And so now I'm gonna show you another diagram next, where I'm gonna show you my hypothesis. So in this diagram is kind of very similar to the right one, except that now we are I'm just showing a little like a smaller window, So you have the epidermis, the mesoglia, the endodermis, with most of the symbionts are, and the mesenterial filament that is located in the gastrovascular cavity with the eggs developing. Next. So the first hypothesis, of course, is that symbionts acquire dissolving organic carbon, and then is transferred to the eggs, and then is translocated to the eggs. Next. But another possibility is that just by feeding on zooplankton, the eggs can obtain carbon that can be passed on into the gametes, or in this case, the eggs. And so these are the main hypotheses that we had. Next. And so with by collaborating with a friend and colleague of mine, Lisa Rodriguez from Villanova University, we designed a pulse chase experiment during spawning season, looking into the physiology of these corals after a natural bleaching event in 2015. And so you can see in the top part, you have the bleach colonies that for both species that bleach and then recover. And then our other group, the non-bleach ones, that they, for the entire time, they did not bleach. Next. And so we did a pulse chase using stable isotopes in the water, but also in the rotifers to test for the acquisition of carbon via autotrophy, but also via heterotrophy. Next. And we specifically designed this pulse chase during the spawning period, which only is 2 or 3 days during the new moon in the summer months. Right? So we started like on day 1, and then on day 3 to 5 was spawning, the release of the bundles, and then day 7. And we wanted to see that because we wanted to catch the bundles and see whether we will find the isotope signature either from the symbiont, right, or the rotifers, to get a better understanding where were they getting the carbon from. And spawning is something you can predict for the most part for this species, but the intensity is what you cannot predict. So that's why it was kind of risky, because we did not know if we were gonna get enough material. Next. And so what I'm gonna show now is what happened with the different groups. So for Montipora capitata, which is a hermaphrodite, what we found for the colonies that did not bleach, what we found is that carbon in the water was incorporated by the symbiont, translocated or incorporated in the host tissue, and then this carbon was allocated to the egg. But we did not see any evidence of the rotifers carbon contributing to the eggs of Montipora capitata. Next. For the corals that bleached, colonies of Montipora capitata, we again see this pathway of carbon incorporation by autotrophy to the egg, but we did not see it in terms of any heterotrophic contribution to the egg. But what we did find, which was very interesting, is we found carbon from the heterotrophy in the symbiont cells. And the next one. For the next one, you see the Porides compressa. And what we found for Porides compressa was really amazing. We saw that again, eggs can obtain carbon from autotrophy, but we also found evidence that eggs can obtain carbon from heterotrophy. So potentially corals that bleach can continue their gametogenic cycle by just feeding more. Next. So as they recover, reproductive processes also can keep going. And oh, sorry, that was in the non-bleach. The ones that bleach, we found mixed results, but overall we did not see this heterotrophy or heterotrophic contribution to the egg, but we saw a lot of the same pattern that autotrophy. So autotrophy was obviously the main pathway for egg development or carbon to the egg. But now we have evidence that for some species, in some cases, there is an allocation of carbon to gametes via both autotrophy and heterotrophy. Next. And that's how some corals can obtain their carbon from. But for some of the corals, they will just need to wait until they recover their symbionts before they can continue to allocate carbon to the eggs. And for the last section, which is the trade-offs in resource allocation, next, what we found in Montipora capitata, here I'm showing you Y diagrams showing the different resource allocation of carbon that we have. So on the top, I have the ones that did not bleach, and on the bottom, we have the ones that we bleached. For the ones that we bleached, the tissue had less carbon. So carbon was depleted by 50% in host tissue of bleached adults, even 7 months after the bleaching event. However, there was 10% more carbon allocated to the eggs in the bleached parents compared to the non-bleached parents. So Montipora capitata is prioritizing reproduction via autotrophy rather than their own tissues. They still did not have enough carbon, but they were prioritizing carbon to the eggs. Next. For Porites compressa is a different story. What we see is that the carbon content recovers in the bleaching adults. So the resource pool or this circle is the same size. So they prioritize adult health maintenance. But we noticed that there was a 45 to 80% reduction of carbon to the eggs in the bleached parents, showing a completely different strategy of resource allocation after a bleaching event. Next. So in this talk, I have shown you evidence of what happens with a coral after they bleach and recovery, especially in the context of reproduction. We went from the reproductive ecology to looking at the physiology using gene expression, and then following carbon to see the exact mechanisms where these eggs were developing, to finally have a better understanding of how these two different species are going to act as they recover and the future generations or the eggs are being produced. And this is really important as we try to better understand what's going to happen with the corals, how are they going to adapt, and how is reproduction and the long term and the future generations are going to be affected by these bleaching events that are now happening more frequently and with more intensity. Next. But I just show you what happened with provisioning, right? And a little bit of gametogenesis. And there are still so many things that we still don't know, especially in the field, and not just in the environment, right? For example, for Porites compressa, it doesn't matter if eggs develop. If there are no males around, there won't be sperm to fertilize those eggs, right? And it's critical that we know if we are losing if one sex is being preferentially bleached and is dying more than the other one. Next. So I think that we have a huge gap really in understanding a lot of just the basic biology and natural history of reproduction in marine organisms. Although we might know or have an idea of how organisms reproduce, for most organisms in the sea, we don't know how often, how much, or if reproductive cycles are even changing. And that's because we don't even have long-term ecological baselines for reproduction. And I believe that this is essential for understanding the long-term impacts of climate change on the phenology and plasticity of reproductive processes. Some corals or some organisms will be able to have a larger window in the context of reproduction, but others might not. And we really need to address and have a better understanding of sexual reproduction in the sea. Next. And so sexual reproduction is important for ocean solutions in a changing climate. And that's because it is the connection to try to understand multiple levels of organization. And sexual reproduction is extremely important for conservation, aquaculture, and wild fisheries. All of these areas are going to depend on successful reproduction, and so that things can be successful, from organism to population to community to worldwide. Next. And now more than ever, I think this is a great time for reproductive biologists to interact with physical oceanographers to try to better understand the reproductive process in the context of a changing environment. We have so many tools now, so many data series that we can use to better understand the phenology and gametogenic cycles, timing of spawning, and whether these reproductive processes are happening or if they are being severely affected or being severely impacted by climate change or other anthropogenic stressors. Next. And we need all hands in deck because reproduction process is a complex problem. We need a lot of hands, right? And not just depend on the scientists, but also the conservation practitioners, the shellfish growers, right? Aquaculturists and aquarists that know so much about how to cultivate and what is required. Citizen science to really bring people in, to really have a better understanding in a larger scale of when things are reproducing and have a more spatial scale rather than just independent smaller studies on reproduction all throughout the world, right? It would be great to have an international spawning network, for example, to see how corals are spawning and how is that affected in the different areas of the world, because in order to understand things such as spawning synchrony, it's extremely hard to do it just by your own or with a small team. So we need all hands in the deck. Next. I was honored to work with Hawaiian school teachers from Hawaiian immersion school programs and integrating them into my research to try to get a larger data set for my coral reproduction work. And this was an amazing experience. And I think that really allowed me to see the power of having a greater team, having a greater community. I organized this coral reef education workshop where we use the bilingual children's book, Kupe and the Corals, that I wrote. And it was so satisfying to work with them about not just the education part, but the research part. And now having a network of Hawaiian school teachers that teach in these Hawaiian immersion school programs about coral reefs and the importance. And I think this was really necessary to obtain more data, but it was really nice in the context of sharing my work and learning from them as well. Next. So ultimately, successful reproduction will be essential for long-term recovery. And I think it's really important to highlight that we really need to know a lot more for most organisms in the sea. And with respect to corals, I think it's really important to take this into consideration, to not only have to decide things such as, what corals are releasing good offspring, where can we locate where are the best mamas, where are the best papas, right? Like what is required, what gametes can we cryopreserve, right? Like we need to start thinking about a lot of this in the context of sexual reproduction so that we can help to not have this pale present next, but we have instead a more colorful present and future for us, and most importantly for future generations. Thank you. And next, and thank you to my colleagues and my funding sources.
Well, thank you. That was terrific. Another great talk, and wow, what a detailed and interesting aspect of coral reproduction, but then also a bilingual children's book. Now you've got me very interested, so I'll have to sort of dig out that as well. We have a couple of questions. Got to keep track of the time, so I'll read them from the chat box. Let me ask you this first one, and it says, Dr. Padilla-Gamiño, do you plan to study the effect of bleaching on the development of the fertilized planulae and polyps?
I mean, yes, definitely. That's the next step, right? So, like I said, like I showed before in my long graph, I focus right now for this in gametogenesis and the development of the egg, because it's actually, that is more understudied than the larvae. Most people tend to jump right away to the larvae and do a lot of experiments with the larvae because they sometimes are easier to catch. Or so yes, that will be the next step. Now that we have a better understanding of what happens in the context of parental transmission, right, and the state of the parent, now it's time to go to the larval, and then the recruits, and then the next generation, and then what happens after the next generation reproduces. So definitely, and those are very interesting questions. And right now there are active labs working on that as well.
Terrific. And let me ask you one more from the chat box. And this one says, how would you account for the difference between the two coral species number of reproductive eggs between the bleached and non-bleached coral samples?
So the number of bleached eggs, let me see if I can read it. Please don't forget that you can. I guess I did not understand it well. So yeah. Montipora capitata is more fecund, right? They release a lot more egg sperm bundles compared to Poraitis compressa. But previous studies have found that Poraitis compressa cellulars or juveniles tend to survive more and grow faster. So sometimes that might be the compensation thing, right? The Montipora capitata knows that they need to send a bunch of them so that they have higher probability of having offspring that can survive, compared than Poraitis compressa that say, well, you know, my cellulars do fine, so I'm gonna invest first on myself and then on reproduction. I think that's what's happening, and that's why Montipora capitata is just investing a lot in reproduction, even though they have not completely recovered 7 months after the bleaching event. I'm not sure if I made a question. If not, you can contact me and we can talk more.
We'll let them do that. Well, we're at the 45-minute point here, so we're going to need to stop. Just say thank you again for your wonderful presentation, and now we'll go on to the next one.
Oh, thank you.
All right. All right, our next speaker is Dr. Melissa Omand. Dr. Omand is an assistant professor of physical oceanography in the Graduate School of Oceanography at the University of Rhode Island. Dr. Omand earned her BSc in physics from the University of Guelph and her PhD in physical oceanography from Scripps Institution of Oceanography. She was also chosen as one of Science News's Scientists to Watch. Dr. Omand's talk today will be Towards a Sensor-Laden Future Ocean. Take it away.
All right, thank you so much, Terry. Thank you to the organizers for inviting me, and thank you to the audience for being here. So I'm going to be talking a little bit about some of the research and technology development that happens in my lab, and then some of my ideas towards how we could move toward a sensor-laden future ocean. Next. All right, so I wanted to start off by highlighting the interplay between the process of making new observations and then the development of new tools or new technology. And I think this interplay is one of the most exciting aspects of my job and in the field of earth science and ocean science in general. So being able to make new observations and have that drive questions about what are the next tools that you need to enhance or deepen your understanding. And then even more exciting when you have a new tool that shows you something that you didn't even anticipate or expect. And so it reveals these new observations. And so this interplay is where I like to put my research at the interface of this. And in particular, I focus on the transport and the fate of organic carbon that is produced by phytoplankton production in the upper ocean, and then the mechanisms by which this is transported into the mesopelagic or the twilight zone of the ocean. Next. So this process is termed the oceanic biological carbon pump. And there are a few mechanisms that by which this occurs. So what's shown in the cartoon on the right side shows some of these different pathways. First is the gravitational pump. This is arguably the dominant sort of form of driver of the biological pump in the ocean. And this is really just the production of organic material in the surface. And then as those phytoplankton die, form aggregates, aggregate and or are consumed by higher predators, and fecal pellets are produced, all of that material tends to be a little bit negatively buoyant and starts raining out, so creating this kind of marine snowfall into the interior of the ocean. The next mechanism is the ocean mixing processes or subduction processes that physically advect carbon-rich water from the surface into the interior. And then the third mechanism is the active pump or the migrant pump, the process by which diel migrating animals come to the surface, often at night, to feed, and then migrate down into the dark interior during the day to seek refuge from visual predators and produce pellets or are consumed by other animals at depth, and thereby creating an active shunt of carbon from the surface into the interior. And I particularly like this diagram because it highlights the interplay between the depth that this organic material reaches in the ocean and the duration that that carbon, whether it's in organic or inorganic form, is sequestered from the atmosphere. And so as sort of a rule of thumb, the deeper that these particles reach in the ocean before being remineralized and converted back into inorganic form, the longer that it stays out of exchange with the atmosphere. And so as you can imagine, there's all these different mechanisms, there's the interplay, there's sort of physical processes, biological processes coming into play here. It's very complex, and it's because of that, it's one of the most uncertain aspects of our ability to predict and model the ocean climate carbon cycle. And so my overarching questions are just looking at sort of what are the dominant mechanisms, what drives the magnitude and the efficiency, so how deep do those particles go, and what are the properties that allow them to reach these really deep depths, and what is the variability over space and time scales. And my goal to address this really variable and sort of diverse process is the development of new tools to really enable us to make distributed, so global scale observations that are persistent and allow us to make these measurements over weeks or months or years. Next slide. All right, so I'll start off before talking about some of the new technology with some of the traditional methods. This sinking or gravitational pump is traditionally observed with something called a sediment trap. And these are fairly simple devices. They are often cylindrical shaped or conical shaped with an opening at the top. And if you play the video there, you'll see an example of marine snow that is just very slowly sort of settling out in the water column and sinking into the opening of these traps, and subsequently that amount of biomass at the base of the trap divided by the area and the duration gives you an estimate of the flux. It's no problem if that video doesn't play. Oh, there we go. So again, you get a sort of visual picture of this kind of gradual snowfall of detrital material. And so one of the challenges with making sediment trap observations is that it requires a research vessel to go out and deploy these and recover them. The durations tend to be on the order of a few days to weeks. and it integrates over that time. So it really just represents one place in the ocean integrated over the deployment duration. So a very sort of costly practice of getting these individual measurements. Next slide, please. And so next, I'm going to describe some of the more recent innovations in terms of making these sinking particle or export flux measurements, namely an optical sediment trap. So this is a essentially what's called a beam transmissometer. So an optical instrument, in this case, gets mounted vertically on the side of a Lagrangian float, such as the neutrally buoyant sediment trap shown in the photograph here. And there's a red LED that emits a laser light over that path length, about 25 centimeters, and is detected. And the attenuation of light and the change in the attenuation of light tells you about the clarity of the water. And when used in this sediment trap orientation, the particles settle on that detector face, and the attenuants, the increase in attenuants over time, the slope of that gives you an estimate of the overall flux. So this is an exciting tool because it now enables a much higher resolution time series than the previous sort of integrating traps, and it can be deployed on autonomous platforms. Next slide, please. All right, so another innovation is the inclusion of a polyacrylamide gel layer in the base of the trap. So what's shown in the foreground of that photograph is a traditional sediment trap. So normally those black lids are open during deployment, allowing particles to sink down inside. And at the base is this 1 centimeter layer of gel that basically provides an anoxic layer. When the particles land in them, they sink down into this gel and their shape is preserved. There's no microbial activity, so the overall material, the amount of material is preserved. And in fact, the DNA and other elemental composition is also preserved. So this is a really exciting tool to really get down into the details of what are the particles that are most important for driving the biological carbon pump and who is producing them, because with these we can begin to sort of trace back the animals or algae that were the original sort of sources of these marine snow particles. Next slide, please. And so I've worked extensively over the years with Meg Estapa and Colleen Durkin, who've really pushed these two approaches forward and have been thinking about how can we merge this high resolution approach with this very detailed particle-centric technique. And so next slide. The solution that we came up with was to use time-lapse imaging. So basically a very low-cost Raspberry Pi camera is mounted underneath the gel layer and is looking upwards through the trap. So that circle represents the column, the cylinder of the trap, and we're looking upwards. And now if you play the movie, you'll see an example of this is just a snippet of time in terms of what we're actually seeing, but you can see the accumulation of particles arriving in the gel. And now we can start to, at a 15-minute interval, we can start to isolate, which particles arrived then, what are their properties, what animals made them, and begin to ask questions about, is there diel variability in the export flux associated with particular migrating animals, or does most of the flux arrive in just this one big snowstorm event, or is it really a distributed, sort of more persistent fallout of particles? Next slide. I'll wait for that one to finish. All right. So one of the challenge with this method was that it sort of gets us part of the way to the goal of being able to make distributed measurements or at least higher, more detailed measurements, but we still are we still have this device on the trap. And so the next thing that I tried was basically taking the camera and getting rid of the trap parts. So now it just is an upward facing camera looking through that transparent surface. That ring is an LED diffuser. So there's an LED potted into the top cap so that we can illuminate the particles that we're collecting. And now when they are deployed on this Lagrangian float by, in this case, by Eric Desaro at UW, they accumulate in this, on this open surface, and we can image them in the same fashion. And one of the really great things about having this type of device is that the float actually came up to the surface periodically, and every time it came to the surface, that open face gets washed off. And so then it comes back down to depth and then goes through another drift and starts accumulating particles again. So we get a reset. And now this really increases the duration over which we can make these kinds of measurements. And so this is actually I'm showing some very, very new data. Most of my colleagues haven't seen it yet either. It's just from the exports campaign from last month. And this is a time series of the Lagrangian float doing its drift and then profile cycle. So on the vertical axis is the depth of the float. The black is the pressure record. So you can see these 12-hour drifts, and then it pops up to the surface, relays data, the snow cam gets washed off, and then it comes back down. And then the red dots represent the time that the pictures were taken. All right, next slide. But to put that into a little bit of context before we look at those photos, I wanted to also highlight the myriad of other tools that are important to sort of to make these kinds of measurements. So this is a sea glider that was following the float over the entire 1-month duration of the cruise. We're looking at the top panel is the upper 200 meters, and the bottom panel is the same data, but going down to 1,000 meters. And the colors are a proxy for the amount of the concentration of particulate carbon in the water. And the black line is the mixed layer depth. And so over time, you can see that in the surface, there was a decrease in the amount of overall particulate carbon, likely the amount of phytoplankton biomass. And at the same time, at depth, you'll see these spikes or this modeling of the carbon proxy, which we think is related to the presence of aggregates. And so around May 16th, there's this front that sort of descends down. Next slide. In the water column, and it makes progress over about 800 meters with about I think about 10 days or so, which translates into a sinking rate of about 60 meters per day. Next slide. And so this is our first clue that something interesting was happening right around when our surface decreased, and now we've got this spike front descending. Next slide. And now we have these circles represent the size of the optical sediment trap proxy, the carbon flux for each of these drifts. And you can see that the large circles, which is on a logarithmic scale, tend to occur right around the same time as we see this descending front. And next slide. And now we can also use our SNOWCAM images to begin to look at what are those particles. And in fact, we do find that these are aggregate particles, really big aggregates. In fact, these the first sequence of 3 images represents this earlier time point, and you can see there's kind of small pellets and things that accumulate over that 12-hour period. But later on, after this export event kicks off, we see a whole bunch of these very fluffy aggregates. And to give some context, that circle is about 5 centimeters across. So these aggregates are, you know, many millimeters, in some cases even centimeter sized particles. So very big on the spectrum. And so this sort of very recent work highlights the value of combining all of these different tools together, and in particular making autonomous measurements. So the weather during this cruise was absolutely terrible. We had so many major storms, and the operations on the boat were really limited, but we were fortunate to have these autonomous measurements that sort of fill in the gaps. And they can, you know, not only fill in gaps during cruises, but also allow much broader and more persistent observations. Next slide. And so I finally wanted to talk a little bit about the development that's actually been the biggest focus of my work over the last few years. And this is taking the same idea one step even farther through the development of what I call minion floats. So miniature isopycnal floats. They're floats that are designed to be very low cost so that they can be deployed in large numbers, and they will vertically seed through the water column, so distribute out so that we can get a sense of what the efficiency of the biological pump is. And this idea was born at a NACFE, a Deep Blue Sea conference supported by the National Academies in 2017. And, you know, the very sort of simple concept was just that we would drop these devices, these little devices off that would sort of drift for some period of time and then pop back up and relay data. Next slide. And now we actually have working minions to play with and drop over the sides of ships and see what happens and whether they come back. So now this is a schematic of the life cycle of our minions. So just like in the original idea, it gets dropped off by a research vessel or any real ship of opportunity. It's very simple to put them in the water. They actually require a lifesaver candy with a preliminary ballast to help them get pulled through the surface layer, and then the lifesaver dissolves and that first weight drops off. And so then they equilibrate at their target depths. So these devices actually don't, and you know, to save cost on their development, they don't have a buoyancy engine, so they don't control their density. We just figure out in advance exactly how dense they are and add just the right amount so that we can get them to hit where we want them to. And if they distribute a little bit vertically, all the better. And so after their duration, they take photographs just like the snow cam. They have other sensors on board. They drop their ballast weight. They actually flip over at that time because the batteries up top are heavier now. And there's a passive traveler inside that slides from one side to the other to make it really stable in this direction. And then there's iridium communication and GPS antenna at the surface for relaying information back to the ship. Next slide. All right, so here's a photograph of the minions. So they're actually made out of a glass housing, and they have their sensors all on one side, and so the other side is actually rounded off glass, just like their predecessors, the Rafos floats. But they're much shorter than Rafos. They're about 18 inches long. And they have, again, that LED ring and the other sensors. Next slide. And so this was kind of an interesting year for everybody. And for me, getting ready with building 20 minions for the exports cruise, a lot of it happened at home. And so this is a photograph of our crib that my 5- and 3-year-old don't use anymore, and it ended up being the only surface in our house that I could use for doing this assembly. Fortunately, with a lot of these, they're small, and most of it is just 3D printed in the internals, and so I was able to do a lot of this without a significant shop. So doing it at home sort of highlighted to me the possibilities for including this in undergraduate education as well, because it ended up being a fairly simple assembly in the end. And the other sort of twist, I guess, to all of this is that my 5-year-old learned a lot more than she'd ever, I ever planned on really exposing her to at this age, about ocean robotics. And so I made this stop motion animation and asked her to narrate it, and she did like an incredible job. So I will play that for you. Next slide. So maybe Clea, if you play this again after, I will narrate it for the audience. All right, so this just shows some of the assembly of the minion. It gets dropped over the side just with a simple slip line release. It sinks down, drops its lifesaver weight, and then stops before it gets to the bottom and takes photographs with its illumination up top, drops the weight, comes back up to the surface, and then relays its data. So I'm excited about the future developments for this technology, specifically the inclusion of a Rayfos style acoustic geopositioning. So we're working with some developers of a miniature hydrophone tag that can be included on the minion and allow us to reconstruct its position during these drift periods. Really essential in the sort of steps towards making these widely distributable. So, deployed in large numbers is the transmission of data. So the onboard processing of these images and the transmission of, the salient information, the metrics that are sort of more compressed than the raw images themselves. And then I'm looking forward to including some strategies for making these longer deployments, maybe including a wiper or some way of clearing the surface off, since these don't profile the way that the Lagrangian float does. And lastly, sort of reaching out to ships of opportunity for achieving this goal of a wide distribution. And I wanted to also mention what I envision as, over the next decade, as we have a lot more autonomous tools in the ocean, not just minions, but all kinds of other devices. I think in order to achieve the kind of coverage that we need, we're going to see not only more autonomous tools, but also more expendable autonomous tools. And I think as stewards of the ocean, as scientists working right at the frontiers of engineering and research and development, we have the opportunity to really push forward some novel materials and approaches and ideas for reducing the impact of these platforms on ocean debris. And so I'm looking forward to participating in efforts to make the minions biodegradable or more ocean friendly, as they, as I hope they'll sort of achieve these really, really large numbers someday. Next slide. And then I wanted to leave you, with a little piece of this vision or a little glimpse into it. This is not minion data that's going to be shown. This is a technology of Apex floats that are using this Rayfos acoustic tracking method. So those yellow dots represent the locations of 4 sound sources. And then if you play the movie, the red dots show the location of all of these Lagrangian floats that are drifting around in the interior of the ocean. And really their GPS, the tracking of these floats is achieved by geolocalizing using these acoustic sources. And so this is the type of sort of breadth and scale and coverage that I hope we'll be able to see with Minion someday, and we'll be able to answer the sort of our questions about the efficiency and the magnitude and the mechanisms and the variability of the biological pump by having this scale of measurement. Next slide. I'm gonna watch it again. I'm glad I made these videos fast. All right, so I just wanted to acknowledge my team, my collaborators, my engineers, Jackson and Aaron, and Tom Rothby, who's just been a pioneer of Lagrangian technology and the RAFOS program, and happened to be my next door office mate when I started my job at URI, and that really has defined and shaped a lot of my interests since. And my other collaborators on the trap work and the exports and Minion COAs, and my funding sources. I'm so grateful to be able to do this work. Thanks.
Well, thank you, Dr. Won. That was fantastic. Again, another example of the frontiers of ocean sciences. That was a really well done. Then I have to say, you know, we've experienced lots of things during the pandemic, and then the repurposing of your children's crib is a great example of necessity as the mother of invention. So kudos to you for getting that done. There are a couple of questions in the Q&A that might be easier. Can you access those, Melissa, as opposed to sort of me trying to?
Let's see.
They should be in the chat, Melissa.
All right. So I see Chris's question, or these are all from Chris. All right, great. So I'll start with the first one. Do you think that SNOWCAM could work with multispectral cameras to gain information about the composition of settling particles? You may be able to use Raman LiDAR for even more detailed spectral information. And then that would be eliminating the signal from the gel matrix. Yeah, wow. I think this would be an amazing thing to do. So using not just an RGB, like a camera that I'm using, like I'm using right now, I do find that even the RGB information has a lot of interesting detail and sort of clues about what these particle types might be. But moving to one with even more spectral range could be exciting. And then Raman would be amazing, and could even allow something like identifying sinking microplastics or other materials that aren't necessarily of just organic form, and isolate those out from the sinking particles. So I think it would be a really excellent thing to do. All right, and the next question is, what is the scale of your current sampling? There are priority regions that you're interested in sampling. I have two areas that I'm really excited about looking at. And so one of them is actually just next week, I'm going to be Chief Sci on a cruise to go and deploy 2 of these RAFOS moorings that are just south of the Gulf Stream, similar to where those 2 yellow dots are. And over the next year, we hope to deploy 2 more. And so this mooring array is supported by the Hui OTZ observation network. And it's a really exciting chance to be able to get that large scale view of what does the distribution of the biological pump look like on a sort of seasonal or on a broad sort of spatial scale. And particularly, I'd love to see if the minions actually can subduct as well. So their name is isopycnal, and at this point, we're not sure how really isopycnal following they are, and how much they're going to really subduct with water as it, as water of a particular density moves and slides underneath water that's less dense. And so if we can evaluate that, then that might allow us to use these as a kind of physical oceanographic metric for the physical pump. So that's, and the Gulf Stream is an excellent place to do that because the North Wall just subducts a huge amount of water seasonally. And so the other area that I'm excited to look at is actually on the submesoscale. So what is the variability of the biological pump and the behavior of the minions in terms of being Lagrangian and potentially isopycnal following on scales of less than 10 kilometers, so 1 to 10 kilometers up to the mesoscale. And in this case, we need really dense observations, kind of, the really edge of this field of submesoscale work is really in some ways getting pushed forward by having drifters, surface drifters that can be deployed in really large numbers to be able to determine kind of coherent structure in the upper ocean and just infer the vertical velocities. And so if we could have a tool that can infer, vertical velocities at depth by, the rate of change of the pressure record in the minion, I think that would be really exciting. So those are the two areas that I'm hoping to work on over the next decade.
Is there one more question in the box?
There is. All right. Yes. Okay. So is there opportunity to tie this into other types of floats and connect with other networks of ocean observations internationally? Have you considered options for linking your research to the UN Ocean Decade? Yes, I hope so. And in fact, tomorrow I'm going to be presenting as part of the Biarritz Imagine Imaging conference. It's mainly European folks that I think are organizing that. And so I'm really looking forward to developing some international connections there. And yeah, hoping to move this. We need global cooperation for any kind of a global problem, right? So if we can have minions deployed by ships of opportunity internationally, I think that would be incredible.
Well, that's terrific. A great presentation, some great questions there. This is very exciting for that. I don't see any more questions in the raise your hand, the Q&A box. So we're coming up to a break.
There's a couple more questions in the chat.
Oh,
on the science
there's a couple more questions.
Side.
All right.
So how long did it take you to do the stop motion video, and will there be a sequel forthcoming? Are you planning to do more videos like Let It Marine Snow, Let It Marine Snow?
Oh, good title. That's a great title, man. Well, now I have to. Yeah, I think the videos are so much fun. It only takes a few hours. You just storyboard it and then make the little pieces. And Antimony also helped me with that, my daughter. And then, I don't know, you just do it. It usually just is like a really late night. And then there certainly will be some sort of a sequel. It's very fun to make these. And yeah, they're easier for me to make than like trying to make some animation with like Illustrator or something like that. So it just works.
I like an example of broader impacts.
Yeah. Broader impacts. I mean, with working with my daughter, but also I think it's maybe an accessible, it makes it a more accessible thing to watch than some of our ways of communicating science.
So I think at this stage, Chris, correct me if I'm wrong, I'm going to thank Dr. Oman again for a wonderful presentation, a great example of sort of the future in oceanography. So my gratitude for that presentation. And my understanding now from the schedule is we're up for a break until 3:45 Eastern time. Is that correct, Chris?
That is correct. That is correct. And we'll see if we can get this video working in the break time.
All righty. Well, thanks, everybody. And now time, take your Zoom break. And we'll see you back here at 45 past the hour.
Hello, everybody. So this is my mommy's minion, and it gets dropped from a ship into water. Deeper and deeper and deeper it goes and goes. And then it gets deeper, and then it stops before it reaches the bottom, and then it takes the pictures, and then it drops the weight, floats back up, and then it goes back up, up. And then there's the data coming from that thing.
That is so adorable. It was worth the wait.
Terrific. And that was very good, by the way. That was so, I'm glad we got that fixed. So, all right, well, we're coming down the second half here. So for that, so let me introduce our next speaker. So the next speaker is Dr. Nikki Trella-Knowles. She's an assistant professor in marine biology and ecology at the University of Miami in the Rosenstiel School of Marine and Atmospheric Sciences. She's also the director of the Moss Marine Invertebrate Collection. She received her BS and MS in cell and molecular biology from Johns Hopkins University and her PhD in biology from Boston University. She did an NSF Ocean Sciences postdoctoral fellowship at Hopkins Marine Station at Stanford University. She is also the founder and director of Black Women in Ecology, Evolution, and Marine Science, and is an advocate for Black women in science and academia. Dr. Trella-Knowles, the floor is yours.
Thank you so much. It's really, really wonderful to be here and talk about some of the work that I've been doing in my laboratory and sort of my future thoughts on where I think things can go with this research. So this is just an introduction to my lab. As Terry mentioned, I'm at the University of Miami. We're right on Virginia Key, right across from Miami City. So, and if you're interested in connecting online on Twitter or via email or checking out our website, please connect. And I just want to introduce who I am because I am more than my my CV. I'm a mom of twins and a wife. So I'm quite busy. My twins are 3 years old and they keep me really busy. I'm a scientist mentor and a teacher. I got into this because I really do enjoy teaching and learning things and exploring. I'm also an advocate. So as was in my introduction, I'll be talking about this more, but I recently started the Black Women in Ecology, Evolution, and Marine Science as a space for helping and supporting Black women in science. And recently I've become the special collections director, which is definitely an area outside of my zone of comfort, but I'm learning a lot and I'm excited about kind of the future of where this work's going to go. So I work on coral reefs, and one of the things that has always drawn me to corals are that it's just a mystery. So I think even if you don't even know what a coral is, if you go snorkeling on a reef and you see all these things, it just draws you in. And so for me, it's a place of exploration, and it's a place of just discovery. So I have always been drawn to coral reefs because there's so many novel things that we can discover. And every time you go onto a reef, you see something new. And then as I started to learn more about their immune systems, I was hooked because it's so complicated. And it's just every day we're just discovering something new, and it totally upends what we originally thought was, the truth. So corals are animals, they're colonial animals. So they start by a single polyp that then settles and secretes calcium carbonate and also clones itself into these colonies. And they provide habitat for fish and other organisms. And they're really characterized, the phylum that they're in are really characterized by having these, what are called cnidocytes. So they're the stinging cells of coral. So if you've ever had the unfortunate event of swimming past a fire coral and getting stung. It's these stinging cells that act like harpoons and have a lot of different toxins and whatnot within them that got you. So, and the other interesting thing about corals is that they have this intercolony connection. So this is a picture or a video here of a coral colony. And then I have zoomed into the structure of the pulp so you can see better what it looks like. And then over here on the right side, what you see is this is a picture of a colony that was cut. And you can see these little, what looks like little wormy things. Those are the mesenterial filaments that are reaching out through, basically it's the guts of the coral reaching through and cleaning the site of this wound and also interacting. So they're really interesting organism because they are clonal, but there is interaction and there's actually a lot of genetic diversity that actually happens within these single polyps. And the other really interesting thing, was also was discussed by Jackie before, was that corals are holobionts, right? So they have within their tissue these symbionts, Symbiodiniaceae, that live within the tissue and provide food and nutrients for the coral host. And in turn, the coral host provides waste that the symbiont lives from. And so it's a symbiotic relationship that's very important for the coral health. But there's also a ton of other players that are found within the tissue. And these are kind of, I would say one of the frontier areas because we're just starting to learn what bacteria and viruses and fungi, microeukaryotes, all these things that are living within the coral tissue. So the coral tissue in itself is its own ecosystem. And the other interesting thing too, from a symbiosis perspective, is that you can have symbioses within symbioses within symbioses, like kind of like the Russian doll comparison. And so corals are an amazing system for wanting to understand symbiosis and also immunity. But coral reefs are under threat. Much due to bleaching, so due to global climate change, as well as due to a lot of local events. And right here in Florida, we're dealing with a major local event where we're seeing that corals are effectively dying at a high rate due to this novel disease, which I'll discuss later. So I often like to ask this, what would happen if corals disappeared? Because as a scientist, I'm really focused in on these kind of granular, small scale things, but sometimes I need to step back and remember why these corals and this ecosystem is so critical, not only to humans, but for biodiversity. So if coral reefs were to disappear, scientifically, we'd have a loss of a lot of unique animals and novel chemical discovery. We'd have no more the beauty and biodiversity and or protection of coastlines, which particularly here in Florida, we really care about. We'd have no more the local fisheries and reduced tourism, right? So there would be a big loss of money economically. And in the end, Florida alone, just Florida would lose billions of dollars. So it really has this economic large scale impact for humans that goes beyond the biodiversity things that drew me into wanting to work on coral reefs to begin with. And so while these reefs, these are pictures here of reefs that have been destroyed, while there are reefs that still exist after a bleaching event or after a disease event, but what you can see is that a lot of these reefs are usually dominated by octocorals or sponges or rubble. And while that can act as a good environment for many organisms, but it's not the same as a coral reef, right? For all the reasons that I discussed before. So one of the questions that's always driven my work is, why do some corals survive and some don't? And I think one area of hope is the immune system. So the immune system is an within humans, we have these two arms of the immune system. It's a defense system, right? And I think recently we've become really familiar with this because of COVID. And we are talking a lot about antibodies and defense, right? But where I'm going to really focus on is the innate immune system. And so this is a system that's less specific, but just as important for, in particular, for coral survival and invertebrate survival, but also for human survival. So the coral immune system can be divided into several sections. They secrete a compound called melanin, which it can be secreted in response to a wound or to a disease, and it acts as a way to encapsulate bacteria and effectively degrade it. Additionally, they can produce fluorescent pigmentation. So this can act as an antioxidant. And in this picture here, what you see, this pink, is from a parrotfish munching on the coral, and it elicits this pink response, this fluorescence response that can act to kill invading bacteria that may be trying to invade due to the wound that was produced. They also have mucosal epithelia, so they produce lots of slime. And within that slime or mucus is a lot of antimicrobial peptides that can battle against different types of bacteria and other invading organisms that may be trying to come in and infect the coral. They also have a lot of genes and pathways, which we know are immune genes based on their gene expression in response to disease and other stressors. And then they also have immune cells. And this is an area that's a little controversial, and I'm going to talk about that because that's where a lot of my work has focused, is showing that they do have putative immune cells. And so I like to throw this picture up. There's a lot on here, and you don't have to understand what any of this is. All that I want you to appreciate are that there's a lot of things here. And this is the coral immune system from a gene level. So we know that corals have a lot of the important immune response genes that we see in other animals. So they have this complex immune system. And one of the things that I'm interested in understanding is, what is it doing? And how do we reconcile the fact that we're seeing so much death to corals with the fact that they have this diverse and seemingly robust and redundant, repetitive immune system? So this is another example of the diverse coral genes that corals have. This is for a gene called tumor necrosis factor receptor. You don't need to know what that is, other than to know that it is part of, it's an immune receptor, right? And so humans have 29 of these. And I thought as a postdoc that surely corals will not have more than humans, and I'll be able to study this gene family and find out all these things about receptors in corals. But at the time, with the available genome, what I found out is that corals actually had 43 different types of this gene. So they have this hyperabundant abundance of this gene, and we don't know why. And so, you know, one of the kind of more specific questions that I like to ask and kind of frames a lot of my work in the background are, why do corals have so much redundancy in the recognition system? So some possible explanations are, they're evolutionarily, from an evolutionary standpoint, they're very old, corals are. And so they've had time to develop this complex system, and for whatever reason, haven't had to lose any of these genes. It's not expensive for them to have them. Or the other kind of explanation could be that they have these complex symbioses. And so they need to be able to know what is friend, what is foe. And that's what these receptor systems are doing. But the problem was, as I was kind of approaching this question, was that how do we test this? And so we kept going back to, well, we need to isolate the cells. But when I would go to the literature, they were saying that they don't have immune cells. So we had to kind of start back and show that they've got these immune cells. And now we're working on further characterizing the functional characterization of these cells. So I'll walk you through some of that work. So the tools that we have for assessing coral health is histology. And these are all tools that we use in human health, right? So histology is a way to look at the tissue and understand the interaction of cells of preserved tissue. We have omics, or in particular transcriptomics. And what this is looking at the DNA. So understanding what the functional units of DNA, like the genes, if they're up or down, and what they're doing. And this can kind of give you this overview of all the different players within the DNA and what they're doing. Additionally, we have enzymatic assays. And this is a way in which this is used a lot actually in disease ecology with corals. And you can use it, it's a method that we use to look at specific types of enzymes that we know are important for the immune response, to see how they are different, and how they respond to different diseases as an indication of immune health. And then recently, my laboratory and other laboratories have been pioneering the use of flow cytometry to isolate and characterize live cells. And in particular, I'll talk about here the immune cells. So flow cytometry is used a lot within the medical field, but also in oceanography. And so we've been taking an approach more based on what we do in the medical field. And the idea is that through using a bunch of different lasers, you can pick up the granularity, the size, the shape of different cell types and be able to separate them out into different groupings. You can also use fluorescent markers to do this, right, with these different fluorescent lasers. And then the other thing that we can do is be able to start to map and understand and kind of tease apart all the different cell types that are within corals, and we can collect it live and start to culture them and examine them. So we've been working on developing this, and this is funded by NSF in collaboration with Dr. Benjamin Rosenthal, who is a researcher at Ben Gurion University in Israel. And this project is led by my student, Grace Snyder. So really, the least understood area of coral immunity are the cells. And so I'm going to walk you through some of the steps that I went through to characterize these cells and how we kind of had to start at the beginning, because we needed to develop tools in order to get to this to a point where we can start to attribute function. So we had to start with the cell culture of these of the coral cells. And so I'm going to just quickly present some of that. And what we did is use this coral called Passalopter damicornis, which is found in the Eastern Pacific, throughout the Indo-Pacific and Eastern Pacific. And this anemone called Nematostella vectensis. And Nematostella is a really great model for laboratory experiments. And it's a really good thing if you're working on corals or anemones and you want an easy model to work on. Nematostella is your animal. So in order to understand and find a good method for culturing live cells from Nematostella and Passalopter damicornis, I had an undergrad working in my laboratory who was interested in cell biology. And so I said, sure, why don't you let's do some cell culture. Here's some methods. We got him trained up. And so he did over 175 different cell cultures. And what we found is that based on all of these, that there was a rough amount of time, around 14 days, between 12 and 14 days for the survival of these cells. And so this was really good to know, because in the future, we want to be able to apply this and use this. And so if we can know how long they can survive, and we can also know that they are really coral cells or anemone cells and not one of the parts of the holobiont, then we can use this technique now to start to maintain and work on the cells in the lab and not have to always focus on getting the whole coral host to do some of the experiments that we want to do. And so then after that, we use the fluorescence activated cell sorting to be able to sort out cell populations. So this is a picture of what an unsorted cells look like. You can see there's a lot of different shapes and sizes and colors, and it's kind of just a hodgepodge of things. So we use the fluorescence activated cell sorting to start to tease apart these populations. So now you can see here, this is the Symbiodiniaceae, that algal symbiont, the dinoflagellate that lives within the coral tissue. And you can see that we found that there's roughly 3 populations of that. And additionally, we looked at the coral cells, and we found that there are 9 other populations that are fairly homogeneous. So then we asked, okay, we have a total of 12 general cell populations, but where are the immune cells? So as a reminder, these immune cells, they are used for, within humans, the way we can think about it is that they're used, for defense. So they go around seeking and looking for pathogens or degraded cells. And as this picture is showing here, this white blood cell going and engulfing this piece of bacteria. And we have all these specialized cells, humans do. And so, and they have different functions within our system. But within corals, we don't really know what they have. So I'll show you kind of what we do know and then show where we've filled in a gap. So there's been this controversy about whether they have these professional phagocytic cells. And often it's been attributed that they only have digestive cells. And so we wanted to test this and look at this. So previous literature has shown in gorgonians, which are octocorals, they're related to stony corals, that they have these cells called amoebocytes that really migrate to the site of a disease and will engulf and interact with particles at the disease site. Within a stony coral, we've seen that there are cells that will migrate to the site of a wound. And this is all done based on histology, right? So this is a dead sample that is preserved, and we're kind of looking at a snapshot in time. But recently, a paper came out through Levy et al. That did a single cell sequencing analysis of Stylophora pistillata. And what they found is that there were 2 populations of cells that had a high amount of genes associated with immune cells. So this gave us good idea that corals should have immune cells. They certainly have the genes that respond to it, but what about functionally? So to cut to the story, we were able to find that they have immune cells, and this is a video here. And these cells, what's so cool is they're able to engulf and interact with bacteria, with fluorescent beads, with dead cells. So they really seem to act like a phagocyte, as we understand, or a macrophage as we understand within humans. And so we're now working on building on this to understand the interaction of these cells with heat stress and other stressors that we see within and on reefs. So I've walked you through some of the flow cytometry that we've been doing and showed you that corals do have these immune cells that we're really excited about and that we're now working on functionally characterizing. And so now I'm going to shift directions and start to work on and talk about omics. So looking at the DNA and the gene expression. And so what motivates this work is that, corals are heavily impacted by disease. So over 50% of the world's coral reefs have died due to climate change and disease. That's staggering. It's insane to me, but that's what we're working with right now. There's an increase in disease virulence and abundance due to human-induced climate change. So we're documenting this and we're seeing this every day on the reef. And this has really posed huge challenges for conservation and restoration efforts. So you can imagine millions of dollars are being spent to conserve reefs and to put corals back out on reefs. And yet, they're still being heavily impacted by disease. So there's a real need to understand how to protect them and what is going on with them. Why are they having so many diseases? Is it something to do with the coral holobiont? Is it something environmental? Is it a combination? And so this is some of the work where my work comes in to understand the immune response of the genes of these corals. But coral diseases are very frustrating to work on. And I'll admit that I actually avoided working on coral diseases for most of my career because we can never tell if what we see in the field is a primary or secondary infection. We often don't know what is causing the disease. We can find correlative evidence, but it's been very challenging to figure out what the exact vector was to cause or pathogen for the disease. The field idea is also very challenging. So different diseases can look the same on the same coral and vice versa. So it's a very specialized art of being able to actually identify them. And often you have to use histology to identify the disease in the end. And then for the pathogen culture has been really challenging. There's a lot of amazing people working on this, and it's just been really hard to be able to effectively culture many of these putative pathogens. And so I have taken the approach to kind of step away from trying to ID the pathogen and instead understand the host response. What is the host doing, and what can that tell us about maybe possible pathogens that may be affecting the coral? So this is an example of the frustration. My student Ben Young made this slide, and I just love it because it shows that for this one disease that's really been studied in Caribbean corals, this is Acropora palmata, it's been studied since the 70s, and 11 different putative possible agents have been identified as being responsible for this disease. So it's a challenging world to deal with, and I am grateful for all of the microbiologists that I work with because it is a hard field to be in. And so given that context of sort of where we are, recently in 2014, it was documented that a disease was noted to be infecting bouldering corals. It started in the summer, and it was correlated with the Port of Miami dredging. It's not sure if the port dredging caused it or if it was just correlative with it, because that area where the dredging occurred, there's also a lot of sewage outlet. The pathogen is currently unknown, and it's affecting over 20 different species of corals. And it started in Florida here and now has spread all the way through to the dry tortugas. So this map itself needs to be updated, and through parts of the Caribbean. And it's spreading fast. So, most corals, once they get this, within it's believed to be waterborne, it spreads very quickly. And so corals can die within a couple days to months, depending on the coral species and the rate of infection of that species. So this is a picture of what it looks like in the field on several different species, and you can see this is the dead skeleton, this is the lesion front here, this white, and the brown here is the putative healthy tissue. And this is a picture of what we do in order to study this disease. We do these transmission assays where this is an infected coral here. Again, you can see the disease lesion. This is the dead skeleton, and this is the putative healthy part. And this is the healthy coral, the experimental coral. And you expose it, and you will be able to transmit the disease. So my group asked, what can the gene expression induced by stony coral tissue loss transmission tell us about this disease? So we set up this experiment. This was in collaboration with Mote Marine Lab and the Smithsonian Marine Station. We set up these transmission assays and then collected these experimental pieces and sequenced them to look at their genes. And for those that are interested, we use 3' RNA-seq. And I'm going to talk about differential gene expression here after this. And so overall, what was really interesting and I think hopeful is that we saw that there was an immune response that was happening in response to stony coral tissue loss disease infection. You don't have to know what any of this is. All I want you to pay attention to right now is this number. So for Orbicella favulata, there were about almost 3,000 genes that were differentially expressed, so either up or down in response to the transmission. And in Montastria cavernosa, we saw about 580 genes that were differentially expressed. And what you'll immediately notice is that they have a very different response, these two different coral species. And we're not sure why, if this is maybe just an issue of how the sequencing was done, or if this is actually meaningful biologically, but we're following up on these experiments to better understand the sort of differential expression that we see. But what was interesting is that there was a lot of immunity and apoptosis, so cell death, and also rearrangement of the cells, indicative of a massive die-off of tissue that was occurring. And this just backs up what we see physically within the corals, right? And additionally, my lab has been taking this a step further to start to understand in incorporating those assays that I told you about understanding the immune cells, right? So what we did is we exposed the healthy part of the coral, so the presumably healthy part, the margin where the disease is. We exposed those to beads and then looked at how much they're able to phagocytose or engulf the beads. And if you see more, that means that the immune system is on. If you see less, that means the immune system is either off or just sort of at a neutral level. And we compare this to controls that were not infected, so another colony, and then ones that were recovered after a week. So one of them had the disease and naturally recovered. And so we wanted to look at how does the immune system react with that. And so what we saw was so interesting and telling and also kind of scary, but something that we want to understand more. So first, what we saw is that the healthy and the margin, so both of these have high amounts of engulfment happening. So indicating that immune response is happening on this whole piece, not just the disease margin, but also through the healthy tissue. So indicating that the whole colony potentially could be having this upward immune response. But when we compared it to the control and to the recovery, we saw this average amount, right? And so this is really interesting and something that we want to explore more, because the good news is that when they recover, the immune response seems to go down. And so one of my hypotheses with this is that the immune response we're seeing is actually having a negative effect on the coral, and that this is being catalyzed by peroxidases. So the frontiers of coral health mechanisms, I think that some of the areas that I'm really excited about are the development of live cell assays to disentangle the immune system function, but also the function of other cells. So the method that I talked about can be used on any cell population. And in fact, my lab is also studying the stem cells and as well as the symbiotic cells to understand the mechanisms by which these cells work. Also being able to develop functional assays for understanding the gene mechanisms in corals, and dissecting the holobiont system in live cells, and being able to come back to that idea of identifying the recognition system within those specific cells. So I'm really excited for the future of this work, and I think we're really going to be able to disentangle a lot of these questions that I've had forever about the function of corals and coral cells. So I just want to quickly shift gears and talk about some of the work that myself and collaborators have been working on in terms of making science more equitable and a more diverse place. So recently, I was part of a paper led by Sarah Davies at Boston University, where we really posed this idea that we need to make science impact more inclusive. And in fact, we really proposed this idea that instead of really focusing on citations that a student may be getting, we actually need to think about this more inclusive language in how we're viewing and how we're mentoring students. Because if we want to maintain and retain diverse students, then we need diverse mentors. And often diverse mentors take this more inclusive lens with their scientific impact, right? And then kind of in my humble opinion, I often get asked, well, then what is required for a diverse science landscape? And oftentimes what I have observed is that there is a lot of monetary support. There are a lot of initiatives for career mentorship, and there's a lot of intellectual support. But I would argue that actually we need to expand this to think about other things besides these three. These are good. We need to continue thinking about these. but I think we need to add in and look at it in a more wholesome way. So including mental health support, checks and balances of mentors and mentees. I think that's one of the problems, is that abuse has been able to continue because there are no checks and balances. Monetary support, confidence, instilling confidence, passion, and exposure within potential mentees, but also within the mentors themselves. Cultural inclusion. And what I mean by this is understanding that if we want to have a diverse landscape within science, then we need to make sure that cultural understandings and engagement and exchange are occurring and are valued, and aren't just sort of a box that needs to be checked. Intellectual support, career mentorship, and peer support is another area that I think is really neglected, and something that I'll talk about briefly, that I've been working on developing more. And then lastly, long-term support. So, you can, if you want to retain and help diverse student population within academia, we have to have long-term support. So it's this sort of multi-pronged thing that needs to be done, in my opinion. And I think we've been doing a lot of work, but there's a lot of work that still needs to be done. So what are some of the personal action items that you can do? Again, these are just my opinions. I think it's good to question your assumptions and beliefs. Many times we react in a knee-jerk reaction and don't actually think about or analyze, well, why am I assuming that? To have a plan for recruiting and supporting diverse students. A lot of times these things are kind of done willy-nilly, and there's not really thinking ahead of how am I going to maintain the student and help to actually change the system. Listen to those who are different from you. Sometimes it may be hard to hear this, but it's important to be able to listen. Make sure to take action and use your power. I think many of the folks on this call have a lot of power, and so use it. And use it in ways to help people. And 5, remember that you're working with individuals. So this is something I always come back to, because oftentimes there's this thing of like, well, let's develop a system and it's going to help everybody. And the thing is that everyone's different and everyone has individual needs. And while there may be some shared experiences that we have, but we are different people. So understanding that, I think, as you're approaching mentoring or initiatives that you're starting, that not everybody is the same. And so on that note, this a year ago, it's like almost a year ago to the day, I started this group called Black Women in Ecology, Evolution, and Marine Science. And I started it because I was really tired of people saying that there were no Black women in ecology, evolution, and marine science. And so I made a call out on Twitter, say, am I the only one? And lo and behold, I'm not. So, the group has now become, we're officially a nonprofit, and we're working on our 503c1 official stance. We have over 200 members that range from undergraduate to all the way through retired professionals. And we have monthly virtual events and socials where the idea is to, one, create community, but also to help with some of the deficiencies or things that have been neglected for many of these women in terms of their career development and for them to be able to advance. And then lastly, too, it's an area too for support. So being able to see other people that look like you, and while they have totally different experiences, but there are some shared things that we can talk about and strategize in a space like this that's so important. So if you or someone you know identify as a Black woman in ecology, evolution, and marine science, please reach out. We're on Instagram and Twitter. We also have our email here. You can contact us, but we also have a sign up page on our website. And with that, I am happy to take questions. I just want to thank my laboratory, and particularly Grace Snyder, for the cellular work that she's done, and my collaborator, Benjamin Rosenthal, and his laboratory for the work that they did. And I'd like to thank all of our funding sources, in particular NSF, for helping me build my, my research laboratory. So thank you.
Well, thank you, Dr. Trillinolles. That was really, again, a very powerful presentation. I think both on the sort of impacts of coral eco-immunity that you talked about, and then finishing with sort of a very strong call to action in terms of diversity and marine science. And I saw your call to action on Twitter, and it was very neat to see it sort of go through. And I love the logo you all came up with, so it's actually really good. For that. So we don't have a lot of time before going to the panel, but I guess a question I'd ask from you sort of is because the senior level marine scientists tend to look more like me than you. So what advice should you have for sort of my generation of folks to sort of help your generation?
I think that it really comes down to action and using your power. So there's a lot of lip service that happens and a lot of uncomfortable conversations that people don't want to have. And the thing is that you just need to sit down and have the conversation and then take action with it. And you know, educate yourself, but also then take the action. There's often a lot of educating and then it stops. And I think the other thing too is to actually see that diverse perspectives are a good thing and actually live that, you know? And so really examining, well, is my lab diverse? And when I say diverse, I don't just mean race. I mean everything, you know, am I judging this with some sort of preconceived notion or bias when I'm, you know, looking at an application? You know, I think little things like that can help to shift that. But it really does take work of allies like yourself to help shift things, because there's only so much that people of color and women of color can do. It's a lot of work.
No, I agree, and I think you're sort of already overburdened, right? And so I think that we need to just do a better job in a real sense. And I really like what you said there. You can read your books, you can take your courses, and you can do your seminars, but you can't stop there.
Yeah.
Right. Action is the key thing. So anyway, thank you so very much. What a great presentation to the group. A good way to sort of end the sort of this year's Frontiers of Talks before we transition over to the sort of panel. So very much appreciated spending time with you today. I know the group is very, very excited to have you. So thanks again. All right. That was terrific. I guess we have a minute or two, but I wanted to make sure that we transitioned in here. Chris, how are we doing?
We're doing well on time. In fact, we're dead on time. That image is coming through a little bit blurry. I wonder if the tech people can help there.
Well, do you want me to try to get started with our last intros for the last section?
Yes, let's get started.
Okay. That sounds good. So again, now we're going to move on to the last part of our program. It's our panel with Symposium alumni, the folks who presented at last year's Symposium, and they'll be sharing with us about their thoughts on the future of the oceans. We will hear from 3 Symposium alumni today, Dr. Hilary Pilevsky, Dr. Philip Bresnan, and Dr. Jess Tierney. After each of our panelists completes their presentation, we will open the session up for questions. You can put things in the chat box, of course, in the Q&A as we're moving along here. So let me sort of move on quickly over to our first panelist today is Dr. Hilary Pilevsky. She is currently an assistant professor of Earth and Environmental Sciences at Boston College. Dr. Pilevsky has a BA from Amherst College and a master's and PhD from the University of Washington. Her research focuses on marine biogeochemistry and the mechanisms that enable the ocean to absorb carbon dioxide from the atmosphere. Her studies combine field measurements at sea, biogeochemical sensor data from autonomous moorings and robots, satellite observations, and global climate model simulations to improve our understanding of the ocean carbon cycle. Dr. Pilevsky, over to you.
Thank you so much. And it's such a pleasure to be back again on Zoom. Thank you again to all of the organizers for making this possible in a fully online format. And posing this really broad question about the future of ocean sciences. So I'll start by first saying I'm really grateful for the vision that Dr. Quinn shared at the beginning of this symposium for the future of ocean sciences. and particularly the emphasis on a vision for more inclusive future for ocean sciences, especially not just for the science itself, but centering the people who do ocean sciences. And that's really been an amazing theme through all 4 of the fantastic talks that we've heard already today. And I also really appreciate the work that NSF is already doing and the efforts, including having this kind of conversation to find ways that we as a community can do better. And having just heard these really 4 fantastic talks, I also wanted to just say how much I've appreciated the exciting work and perspectives shared by our earlier speakers, really groundbreaking science and efforts to improve how we do science, and wanted to especially say how much I appreciated Dr. Pico's work on the history of racism and colonialism in earth sciences, and the perspectives that Dr. Traylor Knowles just shared on an inclusive view of scientific impact and how to support a diverse science landscape and work supporting Black women in marine science. So coming back for this alumni panel, it feels like an especially challenging moment to provide an update on the past year. So I'm going to give it a stab, but definitely this past year has been one where I've focused a lot on reevaluating what parts of my work feel most important to me and are most critical to focus on in a year that probably, like all of you here, it felt totally impossible to juggle all of the balls. And really the summary for me has been that what matters most and provides my strongest motivation in the work that I do, both in and out of my professional life, is the focus not just on the ocean science itself, but on the people who do this work and creating an inclusive and supportive scientific community. So I'm going to talk very little about the science itself and the motivating science questions of my work in this update, and really focus more on some of the things that I've done this past year, focusing on the people that I work with. And also in the spirit of updates on this past year, something that I didn't do in my presentation at the symposium last year and have tried to do more and more over the past year is acknowledge the land that I'm on. Because I live and work on the ancestral and unceded land of the Massachusetts and Pawtucket people here in Boston. And as a white ocean and climate scientist, I think it's really important to acknowledge the role of settler colonialism in contributing to the global climate change we're experiencing today, and that I focus on in my own research and teaching. Next slide. And so first and foremost, and as an update on this past year, these 4 fantastic early career scientists have been the foundation of my new lab group at Boston College. So I here as an assistant professor last year, and this is the end of my 2nd year. And so all 4 of these folks started working with me in the middle of this pandemic last September. So Jose Cuevas and Meg Yoder just finished their 1st year of graduate studies. Stevie Walker just finished her undergraduate degree doing a senior thesis with me. And Dr. Kristen Fogerin is a postdoctoral researcher working in my lab group. And so in thinking about what this past year has meant for people who do ocean sciences, I wanted to note, so just of this example in my lab group, 3 of these 4 folks had plans to go to sea and do field work that were canceled or postponed in the early stages of the pandemic. And all of them have had in various ways to kind of replan what the early stages of their career would look like. Stevie, as an undergraduate in particular, had to completely change the plans for her undergraduate senior thesis, which was going to include field work and lab work, and instead shifted focus to computational work, instead doing a really fantastic thesis that was focused on analysis of CMIP6 model data. And every single one of these folks had to figure out how to build community, how to work with me, how to work with each other in an almost entirely remote format. And so I am amazingly proud of the science that they've accomplished and the work that they've done this past year. And I think there's a really important narrative about the resilience of our early career scientists and scholars, but it's also really important to acknowledge that this has been a tough year that has impacted them and other people at these early career stages in really important and profound ways. Next slide. I also want to talk a little bit about some of my really fantastic collaborators at other institutions. So right at the beginning of the pandemic, we had an exciting project funded by the National Science Foundation to add biogeochemical sensors to a physical transport array in the subpolar North Atlantic. And I'm noticing here that the slide had a little bit of a issue. So you'll see there are a dark green line in the Labrador Sea and a light green line across the rest of the North Atlantic, and they're a little farther south than they're supposed to be. But this is the location of the OSNAP transport array. And so we had gotten this really incredible funding to add oxygen and carbon dioxide sensors onto a portion of this transport array in the Labrador Sea, and also some opportunistic work to also add some sensors in the Erminger Sea with support from WHOI with collaborator Dr. Isabella Labrosse. And so that meant that last summer, with all of the restrictions of the pandemic, we had to figure out how to deploy all of these sensors with all of the restrictions for being able to do science at sea. And so I am so grateful for the work of this incredible set of collaborators led by Jamie Poulter at the University of Rhode Island, and with incredible support from people in many countries across many cruises. Our sensors were deployed, I think, on a total of 4 different cruises from 3 different countries, an innumerable number of emails, Zoom calls, Slack messages to coordinate and figure out how we were going to be able to pull this off. And we deployed 70 oxygen sensors and 6 carbon dioxide sensors through all of this fieldwork last summer. This is a photo shared with me by Sarah Nickford, showing her and Heather Furry, who both supported this project, taken by Jennifer Botrin, showing what it looked like to do fieldwork wearing masks at sea. And so again, really a story of overcoming some of the challenges of this past year, but also the incredible work coming together and extra effort that people had to make going above and beyond. Next slide. And I'll also say in the same spirit, I have been working for the past 5 years with this really incredible program supported by the National Science Foundation, the Ocean Observatories Initiative that maintains these long-term sensor arrays. And so there was also a lot of coordination with the folks who were trying to figure out how to maintain that long-term time series in the context of pandemic restrictions, collaboration with this other program, and again, figuring out how to turn those moorings and continue that time series, particularly the location that I've been focusing on in the Erminger Sea, despite all of these challenges. Next slide. And so a new project that I've taken on in this past year is really trying to build on the importance of that type of collaboration together with Sophie Clayton and Heather Benway. We had at the very beginning of the pandemic proposed to put together a workshop that was supposed to happen in person this past June. That certainly didn't happen, but instead we have convened a working group with 22 really fantastic members, and we're going to be meeting synchronously online later this month to do that work of bringing together people to try to do that community building, developing guidelines and best practices for how we as a community can best use the data that's being produced by the Ocean Observatories Initiative focused on the biogeochemical sensors that I use in my work. And so reevaluating what we were planning to do, and now are planning to have an in-person workshop pushed back a year to next summer in June of 2022. Next slide. And then the final collaboration that I wanted to mention is one that's really focused on teaching. This is a collaboration that had existed prior to the pandemic, actually doing a lot of remote collaboration focused on how to use data from the OOI and bring that authentic data into the classroom, and had very serendipitously developed a lot of resources that were already available online for computer-based data analysis, lessons that can be used in undergraduate classrooms. And so not only were those available for lots of different folks in the community, I had the opportunity to use those in a very challenging year of teaching. Next slide. Where I incorporated data from the OOI, both in an introductory undergraduate oceanography class and in an upper level data analysis class, both of which were contexts where I didn't actually have any opportunity to work with my students in person, but they still had the opportunity to work with these authentic data. Next slide. So I want to conclude briefly with thinking a little bit about some of the still very much in process takeaway lessons that I have from this past year of living, working, and teaching in a global pandemic. First is that these disruptions that we faced over the past year plus to our and our students and our colleagues' ability to focus on science due to systemic racism, personal and family health issues, and grief and loss weren't new in 2020, and they aren't over now. So I think it's really important that we make sure that we give students and colleagues grace when they're struggling, whether or not they're comfortable sharing those reasons. And it means that we need systemic structures that provide for more flexibility so that grace is easier to ask for and also easier to extend. And so that means needing to design our classes, our labs, our graduate programs, and our research projects really intentionally and upfront so that we're prepared to support our students and colleagues through personal and global crises without these being events that push them out of our field. And I really think that we have an opportunity and an imperative right now to design our new normal, centering accessibility and inclusivity. Next slide. So this is really just sort of opening that very big question about what the vision for the future of ocean sciences should look like, and builds on a lot of what we've heard from former speakers. I'll say that when I think about what I want ocean sciences to be, I want it to be a place that recognizes, supports, and celebrates wholeness for the people who do ocean science, beyond just our productivity and our publications, in all roles and in all career stages. I think that especially means rejecting a deficit model when we think about diversity, equity, and inclusion, and the contributions of historically marginalized groups, and instead really values the expertise and the lived experiences of Black, Indigenous, and people of color, the value of disabled and queer and international students and scholars, and the perspectives that they bring to their science and to our community. And that includes taking an expansive approach to interdisciplinarity that goes beyond sources, collaborating with partners with expertise in traditional, local, and Indigenous knowledge, as well as in the humanities and social sciences. And finally, also valuing and supporting expertise and work in education, outreach, and community building, just as much as in ocean science research, very much in the same vein as what Dr. Traylor-Knowles was sharing about that much more inclusive view of impact in the end of her presentation. And I will stop here. And Terry, I'm not sure whether you were intending to ask questions now or wait until you get through the whole.
Hillary, thank you for that. We're going to wait till the end
Okay.
for the questions. And thank you so much for a great presentation. It's great to get the update from you. So let me go on to the next panelist, Dr. Philip Bresnahan. He's currently an assistant professor at the University of North Carolina at Wilmington. He has a bachelor's degree in chemical engineering from Lehigh University, a master's and PhD degrees in applied ocean science from Scripps Institution of Oceanography, the University of California, San Diego. Dr. Bresnahan specializes in situ sensor development and applications for the study of coastal and estuarine processes. I like this part. He tinkers with and invents cutting-edge technologies to study biogeochemical phenomena, for example, ocean acidification, deoxygenation, eutrophication, and algal blooms. Phil, over to you.
Great, thanks so much. Hello, everyone, and thanks very much for the invitation to participate in the NSF Frontiers in Ocean Sciences panel again this year. And thanks to all of you for your time today. Can we go to the next slide? Can go to slide 1. Thanks. So we were provided with the prompt to reflect on the past year, as well as to provide thoughts on the future of ocean science. I'm sure the organizers meant to solicit reflections on our science, but wow, what a year to reflect on. So I also wanted to start with an acknowledgement of the many difficulties of the past year. And as Dr. Pilevsky said, far before the past year as well. I want to try to follow in Dr. Plevsky's footsteps here and those of all of today's stellar speakers, which is a particularly tough thing to do, and discuss the broader picture of ocean sciences rather than delve too much into details of my own research. So I'm going to step a little bit out of my comfort zone in this short presentation to describe where my reflections from the past year intersect with my thoughts on the future. Next slide. I was fortunate to be able to participate in two constructive, important, and ongoing conversations this year. On the left here, we have the URGE program, which was an NSF sponsored and Woods Hole Oceanographic sponsored program, which is called Unlearning Racism in Geoscience. I want to make it very clear, I'm not taking any credit whatsoever for this. This was a phenomenal program put on by the team that you can see here. I was just fortunate enough to be able to participate in this. And I'm going to touch on some of what we discussed in that. I also want to talk a little bit about this amazing book, All We Can Save, which is a collection of essays about the climate crisis and solutions in particular, edited by Drs. Ayanna Elizabeth Johnson, a marine biologist as well, another coral expert, and Dr. Catherine Wilkinson. So next slide. One of the really cool things that I picked up from delving into this a little bit more and participating in some what they called circle conversations around the different chapters in All We Can Save was Dr. Ayanna Elizabeth Johnson's framework for figuring out what you can or should do about the climate crisis. And she breaks it into this Venn diagram that you can see here. What brings you joy? What are you good at? And what is the work that needs doing? Next slide. So for me, the first two, joy and skill set, were fairly easy to think about and figure out. I like playing outside. I like playing in the ocean in particular, but in other parts of nature as well. And when I think about my skill set, I think that I'm pretty good at building robots and Internet of Things connected devices to try to understand more about what's happening in the environment in general, especially in the marine environment and the coastal environment. Next slide. Needed work is harder because you could put so many things in this bubble, but I'm going to choose to highlight a bit of what we discussed in URGE sessions 5 and 6, which are on racism and accessibility, as well as racism and inclusivity. Next slide. For me, the intersection in the middle of this Venn diagram here, you could probably come up with quite a few different ways of thinking about the intersection here. But for me, thinking about my skills, the needed work, what I really enjoy doing, this leads me to accessible, high quality tools for public participation in experiential ocean STEM research. So I'm going to talk a little bit more about that too. Next slide. This also overlaps quite nicely with some of the initiatives that NSF and in particular the Division of Ocean Sciences are thinking about. This recent interview that Dr. Quinn gave in Eco magazine on the Ocean Decade goal of making the oceans accessible and inspiring. This quote here, ocean sciences can be alluring and serve as a gateway for many of those missing millions to engage in science and engineering. Next slide. So as ocean sciences can serve as a gateway into STEM. Next slide. I think that we can take this back even a step further, kind of earlier in the pipeline, if you will, and think about how public participation in ocean STEM or university level participation in ocean STEM, high school level participation in ocean STEM can serve as a gateway into the ocean sciences, which can then in turn, as Dr. Quinn said, serve as a gateway into STEM more broadly. Next slide. Some of the papers that I wanted to highlight, this one I actually showed in my Frontiers talk last year. And so I wanted to come back to this. Field courses narrow demographic achievement gaps in ecology and evolutionary biology. And I highlighted this line at the bottom that you can see here. Together, our findings suggest that increasing the number of field courses and actively facilitating access to students from underrepresented groups can be a powerful tool for increasing STEM diversity. Next slide. But how do we actually think about improving access? So one of the papers that we discussed in URGE, I believe this was in session 6, by Morales and others, promoting inclusion in ecological field experiences, examining and overcoming barriers to professional rite of passage, talks exactly about this same topic. How do we provide that access in the first place? And again, highlighting some of the parts of this that really stuck out to me. One strategy could include broadening what constitutes the field, as suggested by some of these other authors. Field experiences could take place in a lot of different locations. And as Dr. Pilevsky was just discussing, the challenges of COVID really forced us to think about a lot of these things as well. Next slide. I also wanted to touch on what is commonly referred to as citizen science. I know that this is a fraught term, so I'm just going to use the term citizen science, even though I want to just be very deliberate about clarifying that I'm not talking about citizenship in the loaded context that it means in this country often, where they talk about how only a small portion of studies in the field of citizen science involves participants beyond mere data collection, despite the unique and diverse perspectives that volunteers may bring to scientific research. Next slide. So putting the pieces together from these various different papers that I highlighted, I think the story for me and my reflections here and how we look ahead are that we need more ways for more people to participate in more parts of the scientific process, whether they're in the general public or other levels. I want to make it very clear that technology is absolutely not going to solve all of our problems, but I do believe that it can alleviate many of them. And then I also wanted to just say again, I don't consider myself to be an expert in all of these topics, but I certainly think they're necessary. Next slide. Some of the things that I actually was able to do over the past year in the challenging, different system that we were operating in was I taught an oceanographic field methods course, which typically would have been taught quite literally in the field. We actually built these new open source carbon dioxide sensors, and the students were tasked with choosing a field environment of their own choice, of their own preference, and going to that area. Some students went to dorms, some students went to eating halls, some students went to parking lots, some students went out to the beach. And I thought this was a really cool way for students to actually choose their field, what was accessible to them, while making technology with off-the-shelf, low-cost parts and open source technologies. Next slide. And just this morning, I started teaching the Summer Ventures in Science and Mathematics program, which is for highly talented North Carolina students to have a 4-week, very intensive experience in research, giving them exposure to what the research process is. And so started putting together what we're going to be working on into a single GitHub organization and drawing on papers from many others who have done some excellent work in the open source ecosystem. So we have the Liu et al. Paper, Emmanuel Boss's group and Thomas Liu working on a chlorophyll fluorometer. We have some work that Ellen Briggs and I have been doing on open source sea level rise or water level sensors, as well as some others that you can see below here. Next slide. And then I've also been working for the past 4 or 5 years on this project called SmartFin, and we are working on finding new ways to enhance public participation in experiential STEM research. And again, try to connect this to levels other than just thinking of participants as mere data collectors, and coming up with ways to establish partnerships with grassroots organizations, schools, museums, municipalities, and so on. I don't have time, of course, to talk about the details here, but this is something that I've been really excited about for a few years. We've had a few NSF REU students work on this, and we're looking to really push the boundaries of what we can do through, if you will, a convergence approach, and working with social scientists and working with museums and professional educators, as well as folks at these grassroots organizations focused on broadening participation. Next slide. So thanks again, really quite delighted to be here. Really appreciate the opportunity, and I look forward to taking any questions later.
Well, thank you, Phil. Thank you for that update. And it does sound like, despite the pandemic interruptions, that sometimes when life gives you lemons, you make lemonade, and you did a nice example of that with your letting the students choose their own research site. So well done. Let me now move on to our last panelist for the day, and that's Dr. Jessica Turney. Jessica is an associate professor at the Department of Geosciences at the University of Arizona. She's a bachelor's, master's, and PhD in geology from Brown University. She's held positions at the Lamont Doherty Earth Observatory and the Woods Hole Oceanographic Institution before leaving the East Coast for the Desert Southwest for a faculty position at the U of A. Dr. Tierney uses organic geochemical techniques and statistical climate reconstructions to investigate historical climate change, to learn about how the Earth system works, and to try to determine what's in store for the future. Jess, over to you.
Yeah, okay. Thank you, Terry, and thanks for having me back this year again to the event. And yeah, so just to, I think given the other panelists and everything they talked about, I might have changed my presentation a little bit after watching all the excellent speakers and everything. And but I did choose to talk a little bit about how we've kind of limped along with our science. But I will say that just like the other panelists, the challenges of teaching this year really shifted my views about how to teach oceanography, how to teach earth sciences in new and inclusive and accessible ways. And so I share their learning experiences sort of all across the board in that sense. So I did want to sort of update on the project that we have here that's been funded by NSF Ocean Sciences, which is to understand a weather/climate system that is very close to home for us in the Southwest, which is the North American monsoon system. So next slide, please. So the monsoon region is, just to orient you, if you're not used to thinking about the fact that there is a monsoon in North America, it's mostly centered in Mexico, but does cross the border and visit us here in Tucson as well as in Mexico. And so you can see in the blue colors there is just the percentage of rain that falls in the summertime, which is a nice metric showing you the bullseye of the monsoon, kind of down near Mazatlan or in the southern tip of Baja. And so it counts for about 50% of the rain we receive here, but the majority of rain in the states, the Mexican states to our south. And next slide, please. The goal of our project is basically to answer this question: what will the monsoon do in a warmer world? So our ability to predict the monsoon on timescales from daily out to multi-decadal is very poor. It's an organized monsoon system, but our future projections of how it might behave under a warmer climate really differ, depending on what kind of model you use, depending on the resolution of the climate model. And so our way of getting at this question, which is kind of what my group does, is actually to look in the past, to use an understanding of the past changes in the monsoon to understand how it responds to large changes in the Earth's climate system, both cooler climates and also warmer climates. And to do that, next slide, we've relied really intensely on the decades of oceanographic research that have taken place in the Gulf of California by multiple institutions, not just US institutions, but also international cruises and the deep sea drilling program. There's a ton of sedimentary records of climate change that have been collected from the Gulf of California and farther south along the Mexican margin that have been stored and curated both in the ODP labs, but also in university core labs. And fortunately, these sediments have been saved so that we can use them now to actually query the past, to reconstruct the time history of the monsoon, going back thousands of years, and in our case, even millions of years. So next slide. One of the time periods that we wanted to focus in on detail was the last interglacial 125,000 years ago, which is a globally warm period. It was forced by changes in seasonal insolation, solar radiation. So it's not exactly the same as a greenhouse gas driven warming. However, it's our sort of nearest term analog to a warmer world. And the sedimentary cores that are from the Gulf of California are really well positioned to study this period. So next slide. My student, Dervla Megan Kumar, this has really been the center of her PhD research. And I showed this slide last year as preliminary results, which indicate that we found evidence, without going into the details of how we're using the leaf wax isotopes, but we found evidence that the monsoon was stronger in the last interglacial, which is really interesting. Now, because of course of COVID, we were really limited in how far we could go to filling this record out for many, many months. And so like a lot of the panelists, I'm sure, I run a large geochemical laboratory, and because of COVID, we were restricted in what we could do. We were initially shut down for a few months. Then we were open up, sort of one person at a time. And it's only really recently, with everyone having been vaccinated now, that we are back at full steam. And so Dervla wasn't really able to be in the lab as much as she would have liked. The good news is that she's actually almost completely finished this record. The data are so fresh, I didn't even have time to plot them up. And so we actually are close to finishing this sort of monsoonal history for the last 125,000 years, which is really exciting. But in the meantime, we ended up doing a project of opportunity. Next slide. Since Dervla couldn't be in the lab, we decided to try something different. And so we made up a project that she could do for one of her dissertation chapters that did not involve being in the lab and could be done, remotely. And that was actually to kind of take this historical approach and reanalyze all of the existing Eastern Pacific paleoceanographic records. So you can see the dots on the map on the left. Those are sediment cores that have been measured for sea surface temperatures, or proxy for SST, going back to about the last ice age. So there's a lot. And you can see on the right that they come from the whole span of oceanographic regimes that we see in the Pacific, the Eastern Pacific upwelling zone, California margin upwelling zone, and the Gulf of Alaska, and so on. So next slide. So using a particular type of empirical orthogonal function analysis, basically principal components analysis, that's designed to deal with data sets with strong trends, we are able to kind of pull out the major patterns and evolution of sea surface temperatures in a spatiotemporal way in the Eastern Pacific. And so not surprisingly, the main trend that we find is just a kind of monotonic warming from the last ice age moving into the Holocene. And you can see in panel A, the time scale there is in thousands of years, and we're looking at 7,000 years ago to 15,000 years ago. So you can see that slow warming that almost every site feels. And then the secondary pattern reflects millennial scale climate events like the Younger Dryas. And so one really interesting and unsuspected finding was that pretty much all of the sites, as those bubbles there show the correlation between each site and that first PC, all of them, warm coming out of the ice age, which makes sense, except for one that's sitting there on the Mexican margin. And so next slide, when we look at that site in particular, this is a site that we generated in our lab. Actually, an undergraduate student generated this record. You can see it just kind of hangs out. There's not much of structure in the record, pretty much stays around 23 degrees Celsius, 20,000 years ago when there's an ice age, and likewise through the Holocene. And so this is really unusual. There's actually very few sites worldwide that don't respond to ice age cooling. So that led us down a little bit of a treasure hunt. So next slide. So we had some climate model simulations of the last glacial maximum. And we took a look at those to kind of understand what the oceanography of the Eastern Pacific looked like in the glacial times. And the first thing we looked at was upwelling. And so you're looking at here as the simulated change in upwelling. And that red area that's sort of sitting near the dot, which is where our core site is, indicates reduced upwelling just off of what would be today Puerto Vallarta. And so that could potentially have counteracted some of the glacial cooling, but it's still a pretty small feature. So instead we looked at the atmosphere, next slide. And in the atmosphere, we found something pretty interesting, which is that the Eastern Pacific warm pool, which is that sort of blob of warm water that sits just to the north of the cold tongue that we're familiar with, cools far less than the tropical average in the model. And we discovered that that was due to less loss via latent heat. So the trade winds were much less vigorous over this region, And the ITCZ was basically trapped north of the equator, in part due to extreme increases in upwelling and cool SSTs in the cold tongue itself. And so you can see in the precipitation change that there's a sort of blob of precipitation indicating where the ITCZ was in glacial times, sort of sitting over that Eastern Pacific warm pool area and preventing those SSTs from actually getting very cold. And so we think there's actually this ocean atmosphere, synergy that's going on here. And the reason that this is particularly interesting is, next slide, the Eastern Pacific warm pool is a really important region for the climatology of Central America. And it's also just woefully underrepresented in terms of paleoceanography. So you can see this is actually a gap on our map here of all the existing paleoceanographic records. And this region is just not sampled. So this project of opportunity actually has now opened a new door for us, which is to sort of focus on understanding climate in this place that hasn't really received that much attention. And then one more thing I want to mention before I end, next slide, is that NSF has been, I want to thank NSF for being accommodating to pandemic conditions. And because this project got delayed due to COVID restrictions in my lab, NSF was able to give us a supplement in order to extend the project, and in particular, sort of fill out our understanding of the monsoon in recent times in the Holocene. And so with the supplementary support, now I've got support for master's student Kira Harris, who, like many of the other students mentioned by other panelists, originally had a field-based project that got canceled because of COVID, and she didn't have funding. And now she has a project. And again, we're leveraging some historical sediments, I guess, that were taken in the 1980s that collected these beautiful varve sequences from the Guaymas Basin. And from these varve sequences, we can actually put together an incredibly high resolution record of climate and oceanography in the Holocene. So we're excited about that work there. And that was entirely made possible from NSF Ocean Sciences. So next slide, just want to say thank you to NSF for supporting us through the pandemic and all the challenges that have come up, and supporting my students who are working on this monsoon project. Thanks.
Well, thank you, Jess. I really liked your explanation of the project of opportunity, another example of how all of us had to adjust on these things, and the treasure trove hunt, that was also a good one, but showed you how you can, once again, make some progress there. So good for you, and thank you for your presentation. So now it's time for questions. I want to start off with some questions. I want to go to each panelist for their response. Probably start at a high level. And so maybe just go in the order that the speakers spoke. So Hillary, Phil, and then Jess. So I'll start with the question of what are the three most important things the field of ocean sciences needs to be doing over the next decade?
Yeah, you started with the easy one, right? So I think there are lots of different ways to answer that question, but the first thing that I think of is a theme of investing in infrastructure and focusing first on human infrastructure, thinking about what we have to do to support the people who are doing ocean science. A lot of really important things that were raised already today about supporting a more diverse, equitable, inclusive, and just field to support the people in ocean sciences. And I think that means thinking really expansively about what we need to make this a viable long-term career path for people for whom uncertainty and where their funding is going to come from year to year is not a career that they can see themselves pursuing. And so that means thinking about technical staff positions, the kind of funding we support, especially for early career scientists and people in positions of precarity. And I think the kinds of human infrastructure support that Dr. Traylor-Knowles talked about, in that there are some pieces that we've really focused on and recognized, and a lot of pieces that we haven't been focusing on, but are also really important for that human infrastructure. I think there's also pieces of that more in the technical side for the kind of infrastructure for what I think of as like the less sexy science, things like the Ocean Observatories Initiative that I've worked a lot with, and other kinds of like long-term time series monitoring where you need to maintain and calibrate and continue working with things that aren't the exciting, fun new toy, but really require continued effort to keep producing the kinds of records that we need to monitor and understand current and future ocean changes.
Thank you, Hillary. How about over to Phil?
Sure. I don't know if I have three things exactly, but a couple thoughts. And I also wanted to touch on something that Dr. Traylor-Knowles showed, the spotlight approach versus the much broader picture of skills that are actually truly beneficial in thinking about who's participating in ocean sciences. And I guess for me, I've always found it kind of interesting that the broader impact statement ends up at the bottom of an NSF proposal, and it's sort of the last thing that people read. Maybe people highlight it a little bit early on in the proposal, but sometimes that broader impacts or broadening participation plan can kind of fall toward the bottom. And I think NSF has really started to change that approach and think about how to more rigorously evaluate this, more rigorously hold people to the promises that they've made in broadening participation and in broader impacts. But I think that continuing down that path and thinking through some of the things that Dr. Traylor-Knowles showed in that particular image from that paper, providing other resources for folks that are focusing on that sort of effort, or those efforts, I should say, not to make it one particular thing, but the breadth of everything that really goes into mentorship, and the breadth of everything that goes into broadening participation. And then I think related to that, this paper, this article in EOS recently, Reimagining the STEM Workforce Development as a Braided River, by Rebecca Batchelor and others, and how people kind of weave in and out of different career paths, different educational routes, depending on personal preferences, choices, family history, everything that might contribute to the in and out. I think finding other ways to reward that in the academic and research enterprise seems like it would be quite beneficial. So I know that's a bit of a vague answer, but really incentivizing those things, it's something that NSF has the capacity to do. So any ways that would be possible, I think would help quite a bit.
Great, thanks, Phil. Over to you, Jess.
Yeah, I mean, I think in terms of, the goal of diversifying ocean sciences, the geosciences, and including more people, I think a lot about my own undergraduate students and especially their relationship to ocean sciences and oceanography. So they love it. They're extremely excited about it. And I teach a lot of undergraduate students, but they have very few avenues to take the next step. And so, I love the idea of doing things that are not, especially because we live in the desert, we're far from the ocean. We can't do a lot of field-based learning. We do a little bit, but these sort of creative ways that we can bring the field, the ocean, into the classroom, including classrooms that are really far from the ocean, but have a lot of interested students in them. And, I wonder if there's just, what I see is between my students that I teach in undergrad, they're incredibly diverse crew, they're excited about the ocean. And then when I see kind of, our applicant pool that comes into geosciences, there's a huge difference in those two demographics, right? So I think those programs that kind of connect, undergraduate students who are interested in ocean sciences into grad school or industry and positions and jobs in ocean sciences are key. And I think, AGU Bridge is one example of that, but I think if we brainstorm up, if there's a role for NSF in like connecting those dots so that we can keep that interest and keep those students in, in the system, in sort of entering into oceanography, because we're definitely losing people between the undergraduate and sort of the graduate level. The other thing I think that the pandemic really highlighted is, the importance of flexibility, and the fact that NSF did offer some COVID relief initiatives was really important. But as Hillary said, things like this are going to come up again, and it made a difference to be able to, you know, go back and say, okay, we need some more time to do this project and so on. And it makes a difference on the ground in terms of supporting graduate students, supporting the students who are doing the science and giving them support so that they don't have to TA, for example, because the project didn't work out. And so I guess where I'm going with that is, you know, more possibilities for things like supplements, add-ons to grants, longer terms for grants. I mean, we rarely finish things in 3 years, and sometimes it usually takes a lot longer, just even in normal life. And, you know, maybe, and that flexibility is not only, you know, helpful for PIs, again, it really comes back to human resources and supporting the students, because they like to know that they have more than 3 years of support or 2 years of support as well. And it's a better sense of security. So that was a little rambling, but those are the two things that come to mind.
That's great, Jess. No, thank you. And let me just sort of touch on a couple of those, I think, if you allow me. So technical staff is a really interesting one. As you know, many of us who run have laboratories. One of the interesting things that similarities at the university and also at NSF is going up the chain of command and say, no, that many of the technical staff are actually on soft money or supported by awards. And so they're really strongly impacted. The lab is not open, you know, this whole thing. Whereas it's easier to talk about students. It's easier to talk about postdocs and graduates and undergraduate students, but there is this, you know, we try to, and maybe it's the GEO is a little bit different than other directorates, or at Ocean Science a little bit different than other divisions. I don't necessarily think that, but it wasn't the direct resonance that you, if you haven't lived that life of how important it is to generate, you know, analyses in your lab. It was a very challenging environment. And so I thank you, Hillary, for bringing that up, because I think that's an area we need to keep pushing, because again, you're trying to give students a hands-on experience in various, whether it's on a ship or in a lab, you know, technical support or the champions at the university, but probably the most underappreciated folks at the university as well. In my experience, both now in academia, but also at NSF. And then, Jess, I want to go on something that you said about the sort of length of awards. This is another one of those things that's kind of cultural, right? Great, if I wouldn't sit back now, right? I'm going to write a 5-year proposal. I could, but that's not where our culture is at, right? So everything you said is exactly right. We've known that for a long time. So I think as we move forward with the new NSF, I think there's opportunity here to sort of change that, to make that a more common notion, to align, if you will, durations of studies and awards. But it's going to have to, we have to do this to ourselves. Sometimes we're our own harshest critics, right? If you ask for too much money or too many years, you can get criticized. So that's something that's, I think, going to come for that. And then, you know, Phil, you mentioned thing about the braided river analogy. I think that's right. I think we need to scrap the leaky pipeline analogy and get to that more braided river one. So I certainly was impressed by that article, and it resonated with me for that. So we've got, looks like, I think my 7 more minutes left. So now the chance you can, some of you've mentioned it, but if I said to you, what can NSF do to change their practices, make ocean science more equitable and open to potential scientists? You know, so now's your chance to take the gloves off and say, all right, it's just the 4 of us here. So please tell me how we can make ocean sciences more equitable and open to potential scientists.
I guess I can take a stab at this first again. I think the first thing I'd say is we need a lot of voices in the room, and a lot of voices maybe beyond people who have reached the point of being the sort of people you invite to give a talk at this kind of symposium, that really collecting a lot of people's voices. I appreciated what you said, Phil, about not feeling like an expert. And that's certainly what I feel like every time I talk about any of this important work. And with that said, I would say, so I really appreciate that NSF sees itself as being able to set culture and control a lot of funding within a much broader system. But I think it's also important to recognize that funding from NSF is not the only thing that influences culture. And so a lot of what we're talking about are what happens in individual mentoring relationships, what happens in classrooms, what happens in lab groups, what happens in the field. And so NSF can set a tone by saying, here's what we want to fund, here's the kind of work that we think is valuable. But I also see the places that people are leaving are at places where you're applying to grad school, like Just was talking about. Who is deciding to go from undergraduate on to graduate school, and then how are we evaluating those applications? You see the same thing in terms of the experiences people have in graduate school. How does that influence whether they decide to go on and want to be a postdoc and pursue an academic career? I think it's really important that NSF is trying to support postdocs with a new call for postdoctoral research fellows. But again, that's going to be a small minority of the people who are thinking about postdocs, and people are going to know that they're highly competitive, and most folks who apply aren't going to get them, like all of the things that we apply for. And then thinking about the next step of what are academic institutions doing when they write job ads for postdocs, when they write job ads for hiring faculty, and applications, and then what's happening through the tenure and promotion process. And so I think there are a lot of places that NSF doesn't have direct leverage on. And I would like to see what we do as a community to say, where are the places that, that are the highest pressure points that maybe we need trainings and rubrics and different metrics of things we evaluate. And I think NSF is part of the solution in saying, you know, there are certain kinds of misconduct that if you commit this, you know, NSF does not fund you. I think that's helpful. I think there are places where NSF saying, here are the kinds of things, like really prioritizing and rigorously evaluating broader impacts that can help support that, but it's also not the whole solution. And so I think seeing NSF as part of bringing the community together to have these conversations about what we need to do, but not the whole solution, is really important.
Thanks, Hillary. Phil, you have anything to add?
I just want to preface anything else I say with, you know, this is not my exact expertise, and it makes me a little uncomfortable to be a spokesperson on this topic, but I guess just kind of doubling down on the broader impacts and broadening participation. I think that's a lever that NSF can really pull. I think putting more funding into that and making it evaluated quite rigorously is going to continue to enhance some of the work that has been perhaps strengthening in recent years and decades around that. But yeah, just you know, saying something I already said again, I guess, putting your money there.
Thanks, Phil. Jess?
Yeah, I mean, I think the problem is that, you know, the way that we operate and the way that NSF operates typically is that we assume everything is a meritocracy, and that if we put out a call and we put out a program, that everyone has an equal shot at getting an award, and nothing could be farther from the truth, right? So you think about the structures like REU, NSF, GRFP, and postdocs, right? 3 different levels that NSF funds. All of them are incredibly highly competitive. And to the point where, you know, when my undergrads say, I really want to do something with the ocean in the summer, you know, I recommend REUs, but I know that they don't have the grades, right? Maybe to get into it. And it sort of breaks my heart, because I have people who want to do it, and I know that they won't be able to get in. And, you know, same thing goes for GRFP and postdocs. I mean, you only get them when you're sort of the best of the best. And the best of the best is going to be students who are from, you know, wealthier institutions. It's going to be, you know, students who are not from underrepresented groups. And so I think we need to kind of like divorce, I don't know, have a divorce between these, how we run these programs and the assumption that it's just an even playing field, because it's not. And if we can think of ways how NSF can actually target the students who aren't competitive for those things, but who might just need to get a chance, you know, get that experience. For example, if they do an REU program, it could be like this sort of state shift for them, right? In terms of getting interested in oceanography. I can get some students in there who maybe they're like a C student, but they're enthusiastic, and it makes a difference. I mean, it'd be an interesting tack to try that would be different from what we've done traditionally. So I'll just throw that out there as kind of a radical idea, but you know, something to think about in how we offer these training opportunities.
Yeah. Well, thanks for that, Jess. Again, it's another one of those things. It's you know, under the guise of meritocracy, we do certain things, right? And that's where we have to start address all these things, because it's you know, it's the whole thing about having an opportunity, right? Because I have, you know, personal experience where you give people who have quote unquote a low GPA or a modest GPA, but they become rock stars, right? They have all the drive, they have all the intellect, but you know, some knucklehead told me they should take physics, chemistry, and calculus all in their first semester of college, you know, and so they are, right? So we're getting past the time now. I wanted to thank everybody here. I know I've got Alex Eisern and waiting in the wings here. So thank you all for coming back this year and sharing your experience with us. It's always a pleasure to see old friends and colleagues and talk about the last year and update us on the science. So thank you for that. And now to finish the symposium, I'm going to turn it over to Dr. Stacy Aguilera-Peterson, who is the Ocean Policy Specialist in the Division of Ocean Sciences, and she will now introduce our final speaker. Thank you for joining us for this symposium. And Stacy, over to you.
Thank you so much, Terry. It is now my honor to introduce Dr. Alexandra Isern, the Acting Assistant Director of Geosciences at NSF. Dr. Isern has served at NSF in various capacities for 20 years, from the head of the Antarctic Sciences section in the Office of Polar Programs to working in the Ocean Sciences and Earth Sciences divisions as a program director and as section head for surface Earth processes. Her extensive experience includes serving as the Executive Secretary for the National Science Board Subcommittee on Facilities. Dr. Isern earned a Bachelor of Science in Geology from the University of Florida, a Master of Science in Oceanography from the University of Rhode Island, and her PhD in Geochemistry from the Swiss Federal Institute of Technology in Zurich, after which she worked in academia as a lecturer, where her research resulted in many months at sea and in the field. Thank you, Dr. Isern, for joining us today for the closing remarks.
Thanks so much, Stacy. I'm really happy to be here, and I'm extremely happy that I was able to sit in for at least a bit of the afternoon. So it's I really want to thank everyone who was able to spend time today for joining us at the 3rd annual NSF Frontiers of Ocean Science Symposium. As Stacy mentioned, I'm the Acting Assistant Director for the Geoscience Directorate at NSF. For those who are less familiar with NSF, the Geoscience Directorate supports research spanning atmosphere, earth, ocean, and polar sciences. The Directorate provides about 64% of the federal funding for basic research in these areas, mostly at academic institutions, but there's a mix of places we support. Moving on to a bit of the political context going now, and I may be speaking to the very well aware here on this call, but we're in this symposium. But on the 1st of this month of June, President Biden proclaimed June to be National Oceans Month. So it was really nice to get that extremely high level attention from the administration. In his proclamation, he noted that it's imperative that we continue to take proper action to ensure that the ocean continues to thrive. He also noted that the ocean is critical because it supplies us with food, recreation, energy, businesses, jobs, and even provides us with the oxygen we breathe. And again, not to mention the essential role that the ocean plays in moderating our climate, which is obviously critical. President Biden recognized the central role of a healthy ocean in sustaining all of our lives, and that we need to find innovative ways to conserve, protect, and restore the ocean. Here at NSF, we've been celebrating National Ocean Month by highlighting the people and projects that we fund. And as we come to the end of June, this symposium really serves as an excellent culmination, showcasing some of the really exciting, transformative research that we support, but also the broader discussion that we just had of really inspiring an ocean community with the future of the leaders that will take our community forward, and also to have this discussion of the importance of diversity and inclusion in ensuring the robust nature of ocean science. In addition, we're also celebrating the start of the UN Decade of Ocean Science for Sustainable Development. The UN Decade was established to recognize an opportunity to reverse the decline in ocean health while continuing to rely on the ocean for our ever increasing needs, particularly under a changing climate. On World Oceans Day earlier this month, we heard that the UN Ocean Decade has endorsed 5 NSF efforts as official Ocean Decade actions. So we were very excited about that. These 5 programs are: first, navigating the new Arctic, which was one of the NSF big ideas. This program brings together research across the social, natural, environmental, computing, and engineering sciences to inform our understanding of Arctic change and its local and global effects, to really create new directions in Arctic science and technology, and in turn help strengthen the economy, national security, and resilience in this important area. Second is the International Ocean Discovery Program. This is an international marine research collaboration that explores Earth's geological and environmental history and dynamics, using an ocean-going research vessel and alternate platforms that are designed to collect subsea forest sediments and environmental samples. And we heard a bit about that this afternoon. GeoTraces is an international program collecting data on trace elements in the ocean that play critical roles as essential nutrients, act as tracers of processes now and in the past, and also affect organisms as toxic contaminants. We also have the Global Ocean Biogeochemistry Array, or GO-BGC, which is contributing 500 robotic floats to collect chemistry and biology data from the ocean surface to more than a mile deep, which will really allow researchers to transform our understanding of the ocean, predict the global effects of climate change, and also to understand the ocean biogeochemical cycles, carbon uptake, acidification, deoxygenization, and ecosystem health. And finally, we have the Coastlines and People Program, or COPE, which supports diverse, innovative, multi-institutional hubs that are focused on critically important coastlines and people research that is integrated with broadening participation goals. So really in franchising the regional communities into the research program from the start. An important part of the UN Ocean Decade's vision is to ensure more inclusive and participative approaches in designing and executing our science. At NSF, we take equity and inclusivity in ocean science very seriously, and we're working to ensure that everyone, no matter their background, has an equal opportunity to study ocean science. So it was really nice to hear some of the ideas and concepts that came out this afternoon. However, as was discussed here, equal opportunity is not simply saying that we value this and that the door is open, but really being dedicated to bringing down barriers that make it harder or prevent certain groups from participating in ocean sciences. At NSF, we're putting into action activities that we hope will help remove these barriers to equity. For example, we have the Partnership for Advanced Marine and Environmental Science Training for Pacific Islanders that's supporting marine science training and providing resources to 5 minorities serving community college in the Pacific Islands. This really allows the community to gain skills and have the opportunities they need to become marine scientists and really lead action in their communities, which are really, as we know, on the front line of climate change impacts on the oceans and in the coastal zone. Similarly, we have the Lewis-Stokes Alliances for Minority Participation. This program gives Native Americans, Pacific Islanders, and other underrepresented minorities opportunities to engage in field experiences and scientific training. NSF also funds the extremely successful Unlearning Racism in the Geosciences. This program aims to assess where racial injustice exists in the field of geology and geosciences in order to help develop anti-racist structures and policies, and also to aid in providing equitable access to resources. So these and similar programs are working to address injustices, provide equality and equity and opportunities to underrepresented minorities, and allowing them to realize their dreams of becoming ocean scientists of the future. I think we're all aware that the future of ocean science must be one that is diverse, inclusive, and welcoming. Without that, the science won't be as strong as it could be by any means. And so as you have ideas such as were discussed this afternoon, please make sure and reach out and share them with us. I wanted to just finish by summarizing what was presented today. We heard Tamara Pico's work on filling data gaps in historical sea level rise and how predicting future sea level is much more complicated than many realize. She also pointed out some uncomfortable truths about the history of oceanography. We learned about Jacqueline Padilla-Gamiño's studies on coral in Hawaii and how bleaching affect coral reproduction. We also heard about her coral conservation and outreach work, including her book, Coupe and the Corals, which has now been translated into 5 languages. Melissa Ullman, with the help of the accompanying narration from her daughter, which is just fantastic, told us about how her minions helped study the biological carbon pump in the ocean's twilight zone. And Dr. Nikki Traylor-Knowles told us about coral immune systems, some of the frustrations with working on coral diseases, and the need for diverse mentors. She also highlighted the important group, Black Women in Ecology, Evolution, and Marine Science, which I hope will gain some new members from this symposium, and I'm sure we'll be hearing more about this group in the future. So thank you to all the speakers for sharing their findings and insights. It was good to have the panel to discover what the symposium alumni have been working on since last year and their thoughts about the future of the oceans and ocean science. So thank you very much for joining the symposium for a second year and sharing your opinions and ideas. We have to recognize, as come up as a thread through this whole symposium, that 2020 was an extremely challenging year. Many of us are trying to learn from these challenges and experiences and not only build back better, but in the ocean sciences, build back bluer. We do recognize that increased efforts are needed to address grand challenges in the ocean sciences. And importantly, as I mentioned, such efforts will only be successful if we also take action to increase diversity and representation at all levels. So in the future, we look forward to hearing more about the great advancements that all the speakers that spoke today will continue to develop. And we thank you very, very much for joining the discussion today. I also have to thank the behind the scenes team of Lena, Angel, and Derek from our NSF meeting and events management, as well as Stacy and especially Clea from Ocean Sciences. We've learned a lot about how to run these Zoom symposia and meetings, but each time it's always a little bit at the seat of the pants as technical issues arise. So in closing, we hope to see you again next year at the 2022 Frontiers in Ocean Science Symposium. So thanks very much.
And thank you very much for finishing off this meeting for us, Alex. Thank you very much. And thank you everyone for tuning in.